Hi, good afternoon. Welcome to the UWA Institute of Agriculture's 2024 Postgraduate Showcase we call Frontiers in Agriculture. That's what we termed 18 years ago. Before we get started, I wish to acknowledge that University of Western Australia is situated on Noongar land and that Noongar people remain the spiritual and cultural custodians of their land and continue to practice their values, languages, beliefs, and knowledge. It's wonderful to see so many familiar faces in the audience today, and there are more people coming, that's what I was told. In fact, it was overbooked, so please come in. I would like to acknowledge a few names here. Director of the Western Australian Agriculture Research Collaboration, Dr. Kelly Pierce and her colleagues. And if you haven't met her, I encourage particularly the students and early career researchers to meet her at the afternoon tea and later on. UWA Institute of Agriculture Industry Advisory Board Chair, Terry N. Wright, again meet him. He has been leading that group for a while. Director of UWA Data Institute, Professor Michael Small. Former Director General of Research and Industry Innovation at DPIRD, Dr. Mark Sutingham. Former Director General of Agriculture, Dr. Graham Robertson here. Chief Scientist at DPIRD, Dr. Ben Biddle. Then, of course, we have Adjunct Professor John Hamblin, Neil Turner, and many others. I would also like to acknowledge UWA Emeritus Professor Graham Martin, and I will talk about him later. Distinguished guests, university colleagues, media, students, ladies and gentlemen. Today marks the 18th annual Postgraduate Showcase since the re-establishment of the UWA Institute of Agriculture in 2007. Time flies. In the academic ranking of world universities for 2023, UWA was ranked 22nd in the world and number 1 in Australian agriculture. The current ranking will come very soon, very competitive. The UWA Institute of Agriculture has played a significant role in achieving this goal, global reputation, by integrating research, education, training and communication in agriculture and related natural resource management within the university and outside the university. The Postgraduate Showcase brings together some of UWA's best PhD students to share their research in agriculture and related areas. In fact, including those here today, about 140 PhD candidates have presented at our Postgraduate Showcase over the last 18 years. This slide captures a snapshot of those former and some of who have gone to different parts of the world and occupying great success in their careers. So I think it has been a great initiative for students to engage with a wider audience, industry, and so on. This afternoon, you will hear from 7 PhD candidates from 3 schools. So those candidates so you're going to hear about 21 years of work in 1 hour. So I just calculated 3 years multiplied by 7. The presentations are divided into 2 sessions with a short break in between for afternoon tea. I'm very pleased to now welcome our opening speaker, Professor Amanda Davies. Professor Davies is a social scientist with expertise in demography, population geography, rural and regional development. Her research focuses on examining population growth, pattern of demographic change, and rural community development. An applied researcher, Amanda works closely with industry and government and is of Australia's leading public commentators on population and demographic issues. You would have seen quite a lot of articles coming about population, potential growth, the student mobility, and so on. Professor Davies is a former Dean of Graduate Research School at Curtin University. He is currently the head of School of Social Science at UWA, and Amanda's current suite of projects include a national investigation into rural volunteering, an analysis of economic development pathways in mining transition, modeling and projecting changing mobility patterns in Australia, examining the growth of digital nomad population and infrastructure needs. We are working with her to come and do some work in agriculture and rural development areas. And Professor Davies has supervised more than 20 PhD students and is a passionate advocate for the value of interdisciplinary research in underpinning and driving the critical change needed in our social, economic system to bring about a more sustainable future. Please give a warm welcome to Professor Amanda Davies.
And hello. It was warmer in that photo than it is today. It's very cold. So I'm very pleased to be here today, and thank you for the invitation and giving me a few moments to sort of reflect on a number of things. The first thing that struck me in the introduction was that 18 years ago, I myself was a PhD student, so I feel a bit old at the moment, and I'll go through an internal crisis as I think about my age. But in the meantime, I'll talk to you about some of the things that I've been doing since then and reflect on the importance of agricultural research in what we're doing. So I am largely an interdisciplinary researcher, and part of that comes about from my background, and I think this is probably the case for many of you who are researchers, where your own background informs how you then design your research questions and what you're interested in. And for me, I'm a rural kid. I come from a rural background. And that's then led to my interest in rural communities and societies, which subsequently developed into demographics. But to start, I will acknowledge that we are on Wadjuk Noongar land. We are moving into the Noongar season of Makaroo. This is represented by the colour dark blue, which symbolises rain and cold weather. So it's very hard to argue that that's not the season that we're moving into right now. The 6 Noongar seasons reflect changes that occur in our natural environment, and also the cultural practices that fit with that. So there's no separation between natural environment and cultural practices in sort of the Noongar seasons there. It reflects the mobilities and cultural practices that associate with changes in those environments. The Noongar people have storylines that cover vast landscapes, and that ensured that critical knowledge about food and water sources was passed on from generation to generation. So the starting point here is about how do we feed ourselves and how do we keep ourselves alive. And the landscape was fundamentally important to that. And that transitioned over many, many generations, and as our landscapes changed. So as we went through natural periods of climatic adjustment, the storylines also adapted fundamentally around the need to stay alive, which is food and water security. So in our modern society, with all of our technological advancements, food and water security remains fundamental to life, and it remains at the heart of many of our political, economic and cultural practices. So at the very broad scale, we're still looking at how do we feed populations, and at other scales, we're looking at currently how do we feed people in crisis situations, such as in Ukraine and in Palestine. So food and water is fundamentally important to life, and I think you probably are already converts on that. So today the purpose is that we're gathered here to emphasise the critical role that graduate research plays in shaping the future of our food security, driving innovation in industry and sustaining livelihoods in rural communities. And I think it was nice that it was acknowledged that we're going to hear the collective effort of 21 years of research. So graduate research is special because it's intensive. It can utilise new methods and access new ideas. That's really difficult in other settings. So there's a real special place for graduate research. As the global population continues to grow at the moment at an unprecedented rate, the importance of agricultural research and graduate agricultural research, that long intensive research, cannot be overstated. In my own research, I examine population demographics. It is with a very high level of confidence that we can project that over the next 50 years, the bulk of the global population growth will occur in those countries that are currently least economically advanced, that are the most prone to extreme weather events, and we do not know how to project yet those weather events, that currently have some of the least stable political systems, and that are currently have the least internal capacity to feed their growing populations So we know these places are going to be the ones that have the largest populations. For example, you know, we know that China's going to decrease to around about 500 million and Nigeria is going to move into the 900 million. So, sorry, China down to 500 million, Nigeria into 900 million. Much smaller land mass and much lower coping at the moment based on our current knowledge. So that's a huge global challenge and one of which you are all playing a part in solving. All the little pieces add up together. By pursuing advanced studies in agriculture, graduate students are at the forefront of discovering new methodologies, technologies and practices that enhance crop yields, improve soil health, improve business and trade practices and combat challenges of climate change. These innovations are not only crucial in ensuring a stable and abundant food supply, but are also for mitigating environmental impacts of agricultural activities. And furthermore, and this is something that is very, very important and we're committed to at UWA, graduate research in agriculture serves as a powerful engine for industry innovation, and we can document how this happens both in our own local setting and much more globally, how we use graduate in research with our partnerships in industry to really change things. In addition to benefiting food security and industry innovation, graduate research in agriculture has a profound impact on rural communities. By empowering local farmers with knowledge and resources, researchers help create opportunities for economic growth, job creation and community development. And I think you all already know this, but through my own research, I've seen how research that's developed in labs and on research farms at top institutions that many people can't access and is often developed for quite different purposes, feeds into what's happening on ground in these very disadvantaged communities. I've sat with women in the highlands of Guatemala who told me about some of their horrendous histories of hiding in churches to get away from massacres that were happening. They're the survivors. And then they had to rebuild their communities. And they told me about these sort of stories and what they went through. And they showed me how innovations in farming practices that were developed a long, long way away through researchers they had no connection with had transitioned through various mechanisms to them, and also information about local trade systems for agriculture and how this had helped stabilise their economy. In turn, they could then assemble much needed revenue for schooling for their children, and this is only in a sort of period of 3 to 5 years of turnaround. And then they had used that stability in the community to form a women's collective, which they then assembled the funding and the resourcing necessary to open the first women's health clinic that they had. So people for the first time were able to get assistance. This all comes from the basis of stabilising an economy through agricultural research. The social science research then comes in after that. So we're working together in that. I've also been very fortunate to meet with community leaders from a region in Papua New Guinea. And they showed me how 20 years of research into improving coffee production, the quality of the coffee trade partnerships, and the financing schemes around this had also resulted in tangible improvements in the livelihoods. And this was measured through increased incomes, decreased violence against women, and increased child education. So with these things, we're starting to lay the groundwork of transitioning that community and those livelihoods into something better. In this case, it came through interdisciplinary research and research in partnership. And these are sort of the important themes that we see come through the Institute of Agriculture, working in partnership and working for interdisciplinary causes. Through collaborative partnership and knowledge sharing initiatives, knowledge sharing initiatives, graduate students can and have, and we have evidence of, fostered change in sustainability and resilience of rural communities, and it's changing lives. So I think you can all agree with me that what you are doing is very important. Yes, well done you for all choosing to do your research. But what I wanted to say more so, and this comes from a real academic point of view and from the point of view of something I'm working on at the moment, is it's how you're doing your research in partnership with the universities that's unbelievably important right now in our time in history. And I want to take a few moments to explain that. So it's really about how you're engaging through university and industry that's really, really critically important. In my school, the School of Social Sciences, we're working through what I can only describe as a massive project to adapt our teaching and research to the new world of Gen AI. So I returned from my annual Christmas leave at the start of 2023 to something called ChatGPT. I'm like, what is this? Immediately started working with the staff about how are we going to address cheating and the use of ChatGPT. But very, very quickly, the very smart cookies who do computer science and app development and other things generated new apps based on this new technology. And we could start using it for all sorts of different applications. So on the one hand, we're like, no, this is terrible. On the other hand, this is amazing. This is the greatest thing ever. Now, I'm not a computer person. I turn it on, it works. When it doesn't work, I get a new one. And I'm sitting there about midway through 2023 thinking, yeah, I'm going to have to do something about this. So I went and actually got an education in computer science so I can lead the transition that needs to happen in the social sciences. We need to change the way we're doing things and what we're teaching. The part that we need to change is the data analysis and how we teach our students that, because we can now use tools. The bit that becomes much more important is the questions, identifying issues and questions, and communicating that in a really useful and meaningful way. So Gen AI is changing what we're doing and how we're doing it as a society, and it's something that is going to shape what you're doing in the environment you're working in and the power of your research as well. But you have to navigate it smartly, and that takes me back to the how and why you're working with universities. So populations at the moment are being bombarded with a vast amount of information from various sources, leading to a rise in misinformation and fake news. And I did try and generate a video of Travis Kelce, Taylor Swift's boyfriend, to introduce me, but I failed because I'm not very good at computers, so back to that starting loop. But Gen AI, if you're smart with it, you can actually create things that are completely false, completely and utterly false, and that's fun. So it's fun to have a pop star introduce me, right? But it's not fun when it causes people to have massive misunderstandings and breakdowns of trust. And that is the problem that we're facing at the moment. And agriculture and agricultural research is not exempt from this. So obviously it's well known that there's misinformation about climate change. There's also misinformation, and it's growing, about pesticides, herbicides, and other such hot topics. There's a lot of misinformation, and it's happening at speed. The spread of inaccurate and misleading information poses a very serious threat to our society, affecting our beliefs, our decisions, trusts in institutions, and ultimately our well-being. In the face of this challenge, the role of research and knowledge becomes more crucial than ever in combating misinformation and the promoting of a culture of truth and accuracy. Now, I'm sure we've all sat in debates and discussions where someone has a very different point of view from you, and yours is coming from a point of view of knowledge and science. And you're like, well, you've just got that off Facebook. So how is this the same? You need to be able to overcome that. And people aren't just getting it off Facebook anymore. So they're getting their misinformation from what seems to be very accurate sources. So that becomes a problem for us. So how do we know what research is real, and how do we get people to know our research is real and that they should actually use it and trust it? Okay? There are many papers that are being published in journals that appear to be genuine. So this is the bit where we start getting really serious as researchers. At the moment, you'll be accessing, the grad students here and academics, we're accessing journals all the time. How are you working out what's accurate and what's not accurate? Because there's some brilliant fakes out there. So if it's hard for us who have the most knowledge about our disciplines, can you imagine how hard it is for people who don't have all of that detailed and comprehensive knowledge about the development of your discipline? The most powerful tool you have at the moment, given this situation we have, so given the critical importance of graduate research in agriculture for overcoming these huge, wicked challenges, and given the massive problem that we're facing with Gen AI and the tools, and we don't yet have the tools to be able to distinguish between really, really good fakes and truth, the most powerful tool you have is your human connections, your networks and your partnerships. The people you work with will know your research is real. So building that network and that platform is critically important. If you work with industry and community, not only is your research more likely to be able to be directly used by them, they are the best placed people to be able to inform others and other potential users about your end work. So what you're developing for a farmer in Merredin could well be useful for a female farmer in Guatemala. How are you going to get it there? The best way to do that is to work with industry partners and to build your own networks amongst yourselves so you all know what you're doing is real and others know what you're doing is real. It's the only way we can operate at the moment until we can resolve the issue of fake information. So attending and presenting your research at seminars such as this, well done everyone, is essential for letting others know what you're doing and also expanding your own knowledge about what others are doing. While at UWA, to the graduate researchers, whilst you are at UWA, use this opportunity to build your networks. You're in one of the world's leading clusters. This is the time and place to do it. You've got yourself into the best position you can be in. Build your networks. Let other people know who you are and spend your time investing in that. These networks are not only going to become more important as we seek to address the wicked challenges and work in interdisciplinary teams, but they're more important in letting people know what you're saying is useful and it's truthful. So research and knowledge are the formidable weapons in the fight against misinformation and in the fight for societal advancement, equity and justice. So at the moment, I'm very much looking forward to myself expanding my knowledge by hearing from the researchers that we have today at our great institution. And to all of you graduate researchers in attendance, I do wish you all the best in your future, in your studies, and I hope to be able to follow you as you go through your careers. Thank you.
So thank you, Amanda, for that wonderful introductory speech. And certainly I recommend that you come and address some of our undergraduates and masters and PhD maybe once a year. There are a lot of opportunities where the social science interacting with the technical science can. So this is a small portion of just from us.
Oh, I don't know what I did to earn it, but thank you.
Now on to the research presentations. It's my pleasure to introduce our first chair, Associate Professor Abbie Rogers. Abbie is co-director of the UWA Centre for Environmental Economics and Policy and Associate Professor within the School of Agriculture and Environment. Abbie's research focuses on measuring community preferences for environmental management outcomes and then integrating into economic frameworks to improve transparency, inclusiveness, and efficiency in policy and decision-making process. Abbie has worked across a wide range of topics relevant to agriculture environmental policy, including habitat restoration, biodiversity offsets, invasive species, and natural hazard management. She's currently working with the state and local governments across Western Australia as the Premier's Early to Mid-Career Fellow to develop decision support tools for prioritizing climate adaptation responses to coastal erosion. So please welcome Associate Professor Rogers.
Thanks, Professor Siddique. And I'm really pleased to be here today because, as was mentioned, this has been going on for a while now, over many years, and a lot of my PhD colleagues from back in the day benefited from participating in this event, so it's lots of fun. I'd like to now introduce our first postgraduate presenter, Sneha Priya Pappula Reddy, from the UWA School of Agriculture and Environment. I hope I pronounced that well.
Yeah.
Sneha is in her final year of research, focusing on drought stress management, plant-water relations, and proteomics. Prior to her doctoral studies, she obtained a Bachelor's and Master of Technology in Biotechnology from D.Y. Patil University, where her dissertation research focused on the cytological analysis of synthetic groundnuts at ICRISAT in India. In addition to her academic achievements, Sneha has further honed her skills through additional qualifications, including a postgraduate degree in data science from Purdue University in the USA, and a postgraduate diploma in technology management in agriculture from the University of Hyderabad in India. She also held a senior research fellow position for a few years at the Indian Council of Agricultural Research. Her supervisors are Hackett Professor Kadambot Siddique, Dr. Madan Pal Singh, Professor Harvey Miller, Dr. Chalapilla Badravaj, and Dr. Jayan Pang. Today, she'll be presenting on unravelling the physiological and molecular basis of drought tolerance of chickpea. So please welcome Sneha.
So thank you so much, Professor Abbie, for the introduction. So today I'll be presenting my doctoral research on unraveling the physiological and molecular basis of drought tolerance in chickpea. So before I do that, I want to quickly acknowledge my supervisors and collaborators and the funding agencies. I want to mention that my project is a part of Australia-India Collaborative Project, and it's been funded by Australia-India Strategic Research Fund and the Department of Biotechnology, India. I would also like to acknowledge the RTP Fellowship for funding my research. And so we all are aware that the drought stress has been looming over the Australian soils more in recent years than ever before. And this has been reducing the crop yield in several crops. In chickpea, drought stress is leading to the reduction of crop yield ranging from 20% to total wipeout of the crop. So here are some facts about chickpea. Chickpea is the second most important pulse crop, and it provides the affordable source of proteins. It is rich in fiber, vitamins, and minerals. It also promotes the soil health by nitrogen fixation. Now, to talk about the production and economic value, India ranks first in terms of production by producing 13 million tons, followed by Australia, which by exporting most of its produce, almost 90% of its produce, ranks as the highest exporter in the world and generates a revenue of more than $600 million. Now, this figure is expected to escalate in the near future as the chickpea prices has surged beyond $1,000 per ton, as India has suspended the tariff of 60% on the tax. So these are my research objectives: to evaluate the genotypic variability in morphophysiological and proteomic responses of chickpea genotypes under terminal drought, and to study these differences using a much cost-effective and less time-consuming techniques such as digital imaging, and compare them with manually recorded traits. My study also involves characterization of the functional role of stress-responsive proteins and the associated pathways that are responsible for the adaptation of chickpea to terminal drought. But due to time constraints today, we'll be looking at my first objective in detail. So here it is: genotypic variability of morphophysiological and proteomic responses to terminal drought. So these are the 6 chickpea genotypes that I used for my study. First genotype on the left is the donor parent with QTL hotspot region and drought tolerant traits. Then I had the S1 genotype, which is the comparative susceptible check for drought. And the R1 and R2, the recurrent parents, they are the high yielding elite cultivars. So the recurrent parents along with the donor parents were introgressed using marker assisted backcross approach to develop the introgressed lines, which are assumed to have the increased yield and adaptation to drought. So I want to quickly mention that all my experiments were conducted at the National Phenomics Facility located at New Delhi, India. Due to the international border closure and travel restrictions on account of COVID-19, and due to the ongoing memorandum of understanding between University of Western Australia and Indian Agriculture Research Institute. So before I explain my study in detail, I want to mention that introduced this term FTSW, which I used as an indicator of drought stress. So I used the containers for my experiment, where I I carried out the experiment using 20 to 25 kgs of sandy loam soil and covered the soil with plastic beads to reduce transpiration. And all the pods were measured gravimetrically on each day of the experiment. And FTSW was calculated using this formula to calculate the remaining amount of soil water available for transpiration on each day of the experiment. So my experiment had two treatments. One was the well-watered and the next was the drought stress treatment. The first treatment of well-watered plants was maintained at 1 FTSW or 80% field capacity. Then the drought stress plants, for which I imposed the early drought stress at early podding stage. Then at specific points of FTSW, 0.7, 0.5 and 0.3, I made an extensive measure of morphophysiological traits, which we'll be looking at in my coming slides. At 0.3 FTSW, this was the stage where there were visual differences visible between the well-watered and the drought stress plants. So I snap froze the samples, the leaf, pod and root tissue samples for proteomic analysis, and at maturity, I also recorded the yield and yield components. So here's the list of the physiological traits that I measured, which included mostly plant water use, leaf gas exchange, the photosynthetic pigments, relative water content, and the canopy temperature. The morphological studies included the pollen viability and germination, the root growth traits, pollen viability, and the leaf area and biomass. So last but not the least, I also had the, I also had the yield and yield components. So when I started measuring, I first analyzed the plant water use traits, which is an important component for drought. So the straight line represents the well-watered plants, where the FTSW was plotted against the days after sowing, and the well-watered plants were maintained at 1 FTSW on each day of the experiment by rewatering. So as soon as the drought stress was imposed, we had the first group emerging for S1 and R2 genotype. S1 was the comparative susceptible check, as I mentioned earlier, and R2 was the recurrent parent. So this group can be considered as the most non-conservative group, which perished the earliest. Then the second group emerged as the moderate or the semi-conservative group of the introgress lines. Finally, I had the most conservative group with water-saving traits. It is of the D1 and R1 genotype. D1 is the genotype with QTL hotspot and drought-tolerant traits, which I mentioned earlier. So as it is evident that the genotypes have emerged into 3 different groups, so I have picked out one genotype from each of this group, and we'll be looking into their physiological responses in detail. So here's the pollen growth. So the pollen viability and germination percentages plotted against the FTSW. The pollen viability and germination percentage in well-watered plants was at 90%. As soon as the drought stress was imposed, we can see that there are clear differences between the well-watered and the drought stress plants. D1 genotype had the highest pollen viability and germination percentage with nearly 10 to 20% of reduction. And S1 genotype had the least pollen viability and germination percentage with almost 60 to 70% of reduction. Now let's look into the leaf gas exchange. The photosynthesis and stomatal conductant rates were maintained at a steady rate for all the genotypes, although the actual values differed from each other. D1 genotype had the highest photosynthetic rate and stomatal conductance when compared to I1 and S1, even under well-water condition. But when the drought stress was imposed, we can see that there is a decline in the curve, but at each and every specific point of FTSW, we can see that D1 genotype maintained a higher photosynthetic rate and stomatal conductance when compared to I1 and S1 genotype. Now the photosynthetic pigments, chlorophyll and carotenoid contents plotted against the fraction of transpirable soil water. Sorry again. Yeah, so the chlorophyll and carotenoid contents, there's a clear difference between the genotypes in the chlorophyll and carotenoid contents under well-water condition. But when the drought stress was imposed, we can see that the differences have enhanced. The chlorophyll and carotenoid contents decreased throughout the drought stress, but yet D1 genotype had the higher chlorophyll and carotenoids when compared to the S1, which had the 50% reduction in both chlorophyll and carotenoid pigments. Now the root growth. So I captured this picture at 0.5 FTSW for all the 6 genotypes. So the root depth of each genotype, when we see that the root depth of the well-watered plants and drought stress plants seems to be similar. However, we can see that there is a huge difference in the biomass of the plant. So when I went ahead and plotted this on the graphs, it looked like this. So the root length density and dry weight of D1, I1 and S1 genotype, there was a huge difference within the genotype as well as between the genotypes. When the drought stress was imposed, we can see that the root length density and dry weight has slightly increased, but not as much as the well-watered plants. In I1 and S1 genotype, there is a clear decline in root dry weight and root length density. S1 genotype had the least root dry weight with 60 to 70% of reduction. Now this was all about the physiological responses, morphophysiological responses of chickpea under terminal drought. Now to look at the proteomic responses. So as I mentioned earlier that I snap froze the leaf pod and root tissue samples at 0.3 FTSW. So I carried out an untargeted proteomic analysis. So this is the proteomic analysis of leaves, which revealed around 2,600 protein groups and around 60 stress responsive proteins. And the proteins involved in pathways such as photosynthesis, redox signaling, protein metabolism, and stress signaling had the highest significant correlations with leaf photosynthetic traits and leaf physiological parameters. So the proteomic analysis of roots that revealed around 3,600 protein groups and 128 stress responsive proteins. The proteins involved in phytohormone action had the most positive correlations, and the proteins involved in cell wall metabolism had the most negative correlation with root physiological traits. Now to look at the proteomic analysis of pods, which had the highest number of protein groups, around 4,000, and highest number of stress responsive proteins as well, around 4,000. And the proteins involved in vesicle trafficking and photosynthesis had the highest positive correlation with seed yield, and the proteins involved in stress and defense signaling and energy metabolism had the most negative correlations with seed yield. So this intrigued me to go and look ahead at the seed yield of these 3 genotypes. We can see that there is a huge reduction in seed yield when the well-watered plants compared to that of drought stress. D1 genotype with 40% reduction had the highest seed yield under drought stress. S1 genotype had the least with around 80% reduction. So to conclude my study, ICC 4958 is the most tolerant and that best scope for the drought stress because of these reasons. The first being due to its high pollen viability, maintenance of photosynthesis, greater chlorophyll and carotenoid contents. And the second reason is that due to its conservative water use strategy, better root rates, which I mentioned earlier, the root length density and the greater biomass of the roots. And the third and the final reason is that the specific proteins that I just mentioned that promoted the root growth and the pod development of this genotype, so which enhanced its adaptation. So overall, the morphophysiological traits and the proteins played a significant role in the improved yield and adaptation of chickpea to terminal drought. So thank you. Sorry about the slides in between.
Thank you, Sneha. So we have some time for questions.
Sorry, Rosanna, making me run. Great talk, thank you. So that line, that's the one that you said had the QTL hotspot? Is that right?
Yes, yes.
So have you done any analysis looking at the genes underlying the QTL to the proteins that you've identified through the proteomics?
Yes, so that work is still in progress. That's my third objective, which I'm still looking into it. So we are almost halfway there. So there are clearly some differential abundant proteins that are linked to the QTL hotspot region. Yeah, so we are still, the work is under progress.
Look forward to seeing the results. Thanks.
Yeah.
A great presentation, well done, very good methodology. I'm not a gene jockey, I'm down to a soil scientist. Can you just explain the last point, protein that promote root growth and pod development? I couldn't follow that. What do you mean by proteins?
So as I just mentioned, I would like to, review my previous slides. So like I said, these are all the stress-responsive proteins that we revealed in roots, pods, and leaves. So these were some of the proteins that helped under stress conditions, under terminal drought condition in chickpea that we found out. It helped the adaptation of chickpea to terminal drought. So some of the proteins. Yeah.
Thanks. Gauss Azam from DPERD. Excellent presentation. I think you did better than what I did during my PhDs. Just curious, you introduced terminal drought, say, 65 days after well watering to all of them, is that?
Actually, it wasn't in days. I specifically imposed the drought stress at early podding stage of each respective genotype. So these genotype differed in their phenologies. So whenever each genotype reached its early podding stage, so then I took an average of 4 replicates and then I imposed the drought stress.
That's good. The second part is that big difference in root growth and biomass and length and whatnot. I was wondering, at that stage, still plant growing lots of roots?
So as I mentioned, that picture which I showed, that was taken at 0.5 FTSW, which is equal to 50% field capacity. So yes, it is possible that we see that root growth. And I would also like to mention that it is one of the stages where we see the maximum root biomass and root length of chickpea under drought conditions.
Thank you.
Yeah.
Got time for one more quick question, or no? Good. Okay. Excellent. Thank you. That was really interesting, and I think after the summer we've just been through and waiting for the rain to
dry
actually
spell
hit,
of drought.
we can really appreciate the relevance of this sort of work. And like Nancy, I'm not very knowledgeable about genes, but I really like the photographic images of what's happening under the ground there, because I think, going back to Amanda's point, things like that are really good communication tools for people who have no clue what's going on. Thank you very much.
I'll turn it off. I remember. No.
Our next presenter is Dr. Michael Young from the UWA School of Agriculture and Environment. Michael is a farm systems analyst who specialises in whole farm economic modelling to understand, evaluate and optimise farming systems and their management. He completed his PhD at UWA late last year, which focused on identifying ways for farmers to profitably manage weather variation. Michael was awarded the UWA Department of Agricultural and Resource Economics Award for the Best PhD Thesis. His supervisors were Professor Ross Kingwell and Professor Phil Vercoe. And since completing his studies, Michael has moved back to his family farm in Kojonup, where he runs a consulting business and enjoys the country lifestyle. It sounds really pleasant. Today, he will be exploring how a new approach to farm optimisation modelling can enhance strategic and tactical livestock management in WA mixed farm businesses. Please welcome Michael.
Thanks, everyone. So first, I'd just like to start moving that down by acknowledging my supervisors, Ross Kingwell, John Young and Phil Vercoe. They really made the PhD a pleasant journey as it could be. sorry, I don't need it. So I'd just like to show you how I will spend the rest of my talk telling you how I used my PhD to help Western Australian farmers. So essentially developed a new farm economic model and then applied it to West Australian farms to work out what sort of management adjustments farmers can make in response to the unfolding weather conditions to sort of mitigate the negatives. So this is really important because weather variation is one of the biggest challenges facing Australian farmers and causes the most amount of stress to farmers, but it is possible for them to make little adjustments to remedy the weather variation and increase profit in the good years and reduce the negative impacts of the poor years. But it's pretty tricky for farmers to identify what management adjustments to make and how much of the adjustment to make. And that's where the farm modelling is really beneficial. It's not a new concept. It's been around for several decades. The most common example is the MIDAS model, which has been applied by many UWA researchers all the way through to Eastern Australia. It's had a long history of helping policy advisors as well as all the way down to on-farm advice. A really good example is the Lifetime Wool Project. This was a national project that was all about improving ewe nutrition, particularly around, in Australia we typically have a very uneven feed supply distribution. So in Western Australia, for example, we have a lot of feed in spring, but it all dies over summer, and by autumn, early winter, there's a real feed shortage for livestock. And farmers can obviously feed grain to their sheep during this period, but that's pretty expensive. So traditionally farmers have opted to let their ewes lose a bit of condition during that period of the year to save feeding costs. But as we sort of evolved our research, we started realising that underfed sheep got less reproduction, lower lamb survival, and also lower wool growth. So the Lifetime Ewe project was all about working out where that optimal balance is between feeding the sheep and then also that sort of production of the sheep. So they measured lots of different ewes that were fed different amounts and measured their production, and then the farm modelling came in to go, where's the optimum feed target? So that was the balance between your production per hectare, but then also the feeding costs. So this is a really beneficial project for the industry. I think it helped over 4,000 farmers around Australia, and we still talk to farmers today that are implementing the practices that were discovered through this. So it was a great sort of example of where this modelling can be applied and really beneficial on farm. So although MIDAS has had a pretty good life use and been really applicable to a lot of cases and helped our industry a lot, it does still have its limitations. So for example, it's an Excel-based tool, but the model was sort of too big for Excel, so it was split into multiple workbooks, which made it really inflexible. Excel didn't have any version control, so if multiple people wanted to work on the model at a similar time, you basically couldn't, or you could, but not really. This sort of inflexibility made it really hard to go to new regions or new properties to take the model. So to set ourselves up for the next 20 years of farm modelling and overcome some of these key limitations, John Young and myself spent the last few years developing a new model called AFO. It's essentially very similar to same structure as MIDAS. So firstly, you got the inputs and pre-calculations, which define the coefficients and constraints and variables that would represent the farm system. And this is broken into several modules to make it flexible and easy to add new stuff, refine what's there already. The output of this is then sent into a solving algorithm which identifies the optimal management for the farm subject to the constraints. So for example, a variable might be the number of sheep you have, but a constraint is that you have to feed the sheep X amount of feed at different times of the year. And the outputs are then summarised in a bunch of reports that the user can sort of interrogate and to work out why the model is behaving the way it is and what that sort of means to farmers or whatever the purpose of the analysis is. Some of the key improvements of AFO over MIDAS is that it's built in Python, which makes it a lot more flexible. It's got improved version control, so you can actually track your changes as you make them, which makes it really handy if you're working with other people. It's integrated with the cloud, and it's much more flexible between regions, so you can take it to a new property or a new region without, I mean, it's still a lot of work, but not as much as it was before. We made a few improvements to sort of the biology aspects of the model, so it's got improved livestock representation, so we can optimise the nutrition that the sheep are being fed at different times of the year. We can optimise when the best sale time is, improve some of the feed budgeting side of things, so we've got more feed options, so we can do confinement feeding, crop grazing. We've got more pasture options in there, so perennials and annuals, etc. And we added some more cropping rotations and different crop options. And then one of the more major changes that was obviously really important for this PhD was actually the improvement of the risk, so the price variation and weather variation, particularly for this PhD. And then when we got around to actually applying the model, we found that farmers can actually boost their profitability by up to 16% by making these flexible management decisions during the year in response to the unfolding weather conditions. So adjustments from their normal strategy in response to the conditions of the current year. We then sort of dug into this a little bit more, work out, well, what do farmers need to do to actually achieve that 16%? What adjustments am I talking about? So we sort of focused on 4 key decisions with the livestock focus. Firstly, crop grazing. So that is grazing crops early in the season to allow your pastures to be deferred. It does incur a bit of a yield penalty, so it's not necessarily profitable in every year, but particularly maybe a late break or a medium break, it can be quite beneficial because it allows your pastures to get off and away. And this was worth $27,000 for the case study farm in the Great Southern. We then looked at sort of a dynamic pasture grazing strategy, so we compared a more typical sort of set stocking rate grazing management where most of your paddocks are grazed at a sort of a similar intensity all year round, with a more dynamic strategy where you adjust your grazing strategy or grazing management in response to the weather conditions and how your pasture's growing. So that was worth $44,000, so quite a profitable adjustment for producers to make. Then looked at rotation adjustments, so making adjustments to your rotation choice in response to basically the start of, all you really know when you're putting your crop in the ground is the early rainfall, so making adjustments based on that early rainfall. So if it started off favourably, the year's had some early rainfall, maybe not like this year, you might put an extra paddock of canola in, pull a paddock of pasture out. And this was worth $28,000. Then looked at adjusting your livestock sale time based on the unfolding weather condition again. So that would be like, for example, you've had a pretty favourable start to the year, you've scanned your sheep, and then instead of selling your strategy might be to sell your dry sheep straight away, but in a favourable year you might retain them a little bit longer, get a bit of wool growth off of them, feed them up a little bit, sell them for a higher profitable, for a higher value. So that was worth $14,000. So there's quite a bit of money to be made up there from these sort of adjustments in the year. And farmers are already probably making 50% or maybe 30% of them, but we do believe there's still a lot of room for farmers to make a bit of improvement, particularly around that sort of dynamic pasture grazing. And it's tricky to do on the farm, but the money's there. So as we start facing more and more climate variation, these tactics are going to become more important. But I think the outputs of this PhD sort of extend beyond those simple findings as well. AFO's is being widely applied and still developed in the ag industry. And we've got students that are using it as well. So I've got a student at Murdoch who's currently using AFO to evaluate lupin fodder crops in the Great Southern. And I've been able to use the skills that I learned in my PhD to start my own consulting business, where I probably spend half my time working one-on-one with farmers, sort of evaluating their strategy, working out what adjustments they can make, and then the other half of my time working with industry, like DPERD and CSRO, evaluating the economics of new innovations and technology and working out how that applies on farm. So overall, the PhD has been a really great journey for me, and hopefully I've been able to show you today a bit about how we use the PhD to help Western Australian farmers make those better, flexible decisions as the year unfolds. Thank you.
Thank you, Michael. It's really great to see as well that there's still a continued focus on developing some of these tools through the Agriculture and Resource Economics group, because it's been a key strength of theirs over decades now. So well done. Questions?
I'm pretty loud, I don't need mine. Thank you for a great presentation with an immediate practical application, which is really great. Again, I'm not an elegant economist. I'm not asking any question, just a comment. I'm not sure why you started your presentation with a statement, not a novel concept. Because to me, the concept may not be novel, but the application is such a novelty. So my suggestion is, don't start a presentation, because it is having great novelty. May not be novel to economists, but someone outside, it's a great novelty. Well done.
Cheers.
Anyone else?
Was that valued per farm or per 100 hectare, or what was that?
How big was the farm?
Yeah, the amount you put on.
So that farm was, so that was a 2,000. So the results that the dollar figures for a farm per year, and the farm was a 2,130 hectare farm, so sort of typical for it was an average farm for the Great Southern region.
And there was a question down here.
I just wondering, because you said your model is very flexible. I can tell it's very flexible, but my question is, does the farmers need to know a lot of things to use your model?
Yeah, so there is quite a lot of inputs that go into it. Some you can have at a regional level is satisfactory. Some, if you were to go to a particular farm, you would need their sort of, because each farm is quite different. In this example, we just used a sort of an average farm for the region. But if you wanted to take this model, which we do in the private side of things, to a farm, you need to know what sort of arable area they've got, what areas they've got, because a farmer might be good at cropping or might be good at livestock, so you need to understand where their skills lie, what their production is, what sort of fertilisers they're putting on. So there's a lot of information, but most farmers have pretty reasonable records of that, and where they don't have the data, then that's where you go back to what the research level or the regional level data. If they don't, like pasture growth, for example, is a bit more tricky. Every farmer knows their crop yield, but less farmers will know how much their pasture is growing at different times in the year at different grazing intensities. So that is where you go more to a even simulation model like AppSim or AusFarm or something like that to help.
All right. Does your model also depend on the weather forecast in WA?
It doesn't use so it actually goes, it's more of this is a discrete stochastic model. So you will say, okay, because every year can be a little bit different, but that gives you a million options. So to make it actually feasible to run and actually look at the output and make some sense of it, which is what we did here, you sort of group every possible year you group similar ones together. So for example, in the Great Southern, we said, what are the most important parts of the year? So the break of season, the time of the break of season, whether or not it's sort of like the quality of the break, and then the spring conditions. So they were the three things that we said are the most important. So then we look back at the last 50 years of data and said, okay, which category do you fall into? So if you started off good or at your early break, then did you have a poor spring? So you group all the years together and work out what the probability of those different weather years are, and then that's what goes in the model. So it's more of you're putting in the probability of these different years, but you can't make a decision. So right now, I don't know what the spring conditions are going to be this year. So if I'm putting my crop in right now, I've got to go, well, I know how the break was maybe, but I don't know what the future conditions are. So you have to make that decision based on the probability that it might be 40% of a good spring and 60% of a bad spring. So it's complicated. I don't know if I explained that very well, but yeah, if we can chat more if that wasn't clear enough.
It feels like it's very hard to be a farmer as well. Anyway, thank you.
I think we've got one more question.
Thanks, Michael, for your presentation. Obviously, things on farm and technology are changing rapidly. And I'm just thinking, you mentioned about pasture growth and using drone or satellite imagery to assess pasture growth as a real live sort of update into the system. Have you considered sort of integrating those sorts of factors? That would make it very farm relevant. Even, farmers are keeping a lot of data, say in databases, various databases that they use for farm management, to be able to integrate data seamlessly into your system just to make it sort of much more, I suppose, up to date, rather than having to put in all that data.
Yeah, and it's like pastures from space, for example, is a reasonable tool, and it's only a WA tool, but it's still a good one that sort of measures theoretically every day or whatever it is, every 7 days, taking pasture growth and food readings. Currently it doesn't automatic, so you'd get the data off of pasture and space, put that into a spreadsheet, do a few more calculations, and then put it into AFO. It's not quite a seamless process, but it's definitely something that it's thought about, and it'd be something that would be nice to do, to have it all just, automatically come in every year. You pull in the most up-to-date data kind of thing, or add to what you had last year, but not automatic just yet.
And I think that's great that you've sort of got that focus on usability and flexibility, because you can't undermine the importance of those in any decision support tool. So thank you very much. That was an excellent presentation.
Cheers. Thank you.
Now, our third presenter for the afternoon is Jessie Weller, also from the UWA School of Agriculture and Environment. Jessie graduated with a Bachelor of Science with first class honours in Environmental Science from UWA in 2021. Her honours investigated the effect of soil stresses on the decline of Norfolk Island pines, specifically the ones at the Somerville Auditorium at the top of campus here, and more recently at Cottesloe Beach. Jessie's research endeavours to investigate the modern day structure of deeply weathered landscapes, including sediment layering and thickness, bedrock depth and landform distribution, and how such structural heterogeneity can provide glimpses into its evolution and influence modern-day future dynamics, including nutrient cycling, the water cycle, and land degradation due to human disturbance. Her supervisors are the head of the School of Agriculture and Environment, Associate Professor Matthias Leopold, and Associate Professor Sally Thompson. And the title of her talk today is Geophysical Characterisation of a Lateritic Hill Slope in the Avon River Critical Zone Observatory. Please welcome Jessie.
So thank you for that. Before I get into why subsurface structure is important and my investigation, I would like to thank the Geological Survey of Western Australia for loaning their passive seismic devices to my investigation, and my three supervisors, Matthias, Sally, and Sarah. So I want you to imagine that you're standing in the middle of a landscape that you operate in. You may see that there's small hills capped in native vegetation in front of you. You may see a seasonal stream or a salt lake to your left. You might see an endless sand plain to your right, or rock outcrops behind you. You notice that this landscape is made up of a mosaic of surface features that are variable in their composition and their nature. But what of the surface beneath your feet? Well, the varied structure of the surface, so these surface landform features, are actually indicative of a varied internal structure in terms of chemical composition, physical characteristics, and biological activity. So understanding the interactions between these processes is challenging to earth sciences due to the difficulty in accessing the deeper subsurface. Addressing the complexity of these interactions between physical, chemical, and biological processes is central to improving our knowledge on hydrological dynamics, the downstream delivery of water to humans and ecosystems, the fate and transport of nutrients and pollutants, mineral microbe interactions, and the terrestrial carbon cycling. So all this actually helps us in understanding the overall biogeochemical cycle, land evolution, and land use planning, which can help us in our natural resource management. So to understand and quantify the modern day structure of our subsurface, we do need to look back in time to see how it has evolved. So going back 60 million years, our climate was wetter and drier than it was today. And we saw broad river valleys, much like the Amazon River, cutting into and weathering pre-existing material. And this saw the enrichment of surface material in iron and aluminium. And over time, moving into the Pliocene, this weather became drier and more seasonal, which saw to these surface material rich in iron and aluminium forming crusts due to that evaporation of water. And then in this time period, because rainfall was reduced, we see that erosion became the dominant process, forming landscapes. So this erosion removed all the soft material away from these hardened Jura crust material, which are iron-rich and our aluminium-rich crusts, and this saw to landscape relief inversion. So the landscape we see today, the highest points were actually once the lowest points in the landscape. So they were once our stream beds. And this has implications for our farming and our agriculture because these stream beds are now capped in this Jura crust material. So our resulting landscape is a dissected landscape with small isolated hills capped in this Jura crust material. And these are typically our lateritic profiles, which are comprised of a thin soil material above a 2 to 4 meter thick Jura crust cap. Below the zone is two distinctive layers, our mottled zone, which has speckles of iron oxide, and then below this, our pallid zone, which is our intensely weathered material, which is depleted in macronutrients and often enriched in clay. And then below this, we have our saprolite, which is much like our granite, but it is more weathered and friable, so it is soft. And then bounding this profile is our fresh granite bedrock. We have also seen in the last 100 years or so that human disturbance is changing the structure of our landscape. So the removal of our deep-rooted native vegetation for our shallow-rooted crops has seen the lowered consumption of our groundwater table, which has seen it to rise. And unfortunately, this rise has brought the salts stored over millions of years from in the deep subsurface to the top of our profile, which increases our risk not only of seasonal waterlogging, but of salinity. And we also see that this rise in groundwater table has brought clays to the surface, which can increase our risk of hard pan formation. So this brings us into our study and why we wanted to complete it. So we wanted to try and find a cost-effective means of investigating this subsurface structure, so in terms of sediment layering, sediment thickness, and overall depth to bedrock. We wanted to see if the devices that we picked can allow us to make inferences on the physical and hydrological properties of each depicted layer. So we did this at the UWA farm in the Wheatbelt district. So this area is characterized by a typical lateritic profile, which is common to the southwest of Western Australia and which are underrepresented in subsurface studies. This site also has a very low relief. So our hilltop is 350 metres above sea level and it goes down to 305 in our foot slope area. The climate is Mediterranean, so 50% or more of rainfall falls in winter, which has implications for our seasonal waterlogging and that movement of clay in the subsurface. We also see that this site has been disturbed by humans. So in our hilltop, we have our remnant eucalyptus species, and then in our foot slope, this has been replaced by shallow-rooted pasture grasses. The instruments that we decided to use and compare were geophysical instruments, which investigate variations in our subsurface based on physical composition variations. So the first instrument was our electrical resistivity tomography, and this instrument actually measures the resistance of each layer to an electric current that is actively injected into the ground. So if this electric current encounters a layer that's rich in clay or is water saturated, these layers will be less resistive to that electric current compared to a rock-like material. Our second instrument was the passive seismic, which measures ambient noise contrasts in the subsurface. So as these ambient noise moves from the subsurface to the surface, it encounters different layers of physical characteristics, and it distorts that noise. So if you've got a layer rich in clay or water saturated, it will actually slow down this velocity of ambient noise compared to a rock-like material, which will speed it up. So we want to look at the results individually across the two instruments. So firstly, the electrical resistivity tomography depicted 3 heterogeneous vertical subsections, which align nicely with landscape position. So we've got our hilltop, our mid-slope, and our foot slope. And then our hilltop and foot slope, we see a 4-layered subsurface structure. So in the hilltop, we see a very thin, high resistivity layer, which we attribute to that hardened Jura-crust material. Below this layer is our pallid zone, which has a low resistivity, which is attributable to either high clay content, high salinity, or water saturation. And then below this zone, we see our saprolite layer, which is our thickest zone here. And this layer has an intermediate resistivity, which stems from it weathering from the bedrock below, which has a high resistivity. In our mid-slope, we see a 3-layer subsurface structure where our pallid zone actually reaches the surface and is exposed there due to the movement of wind and water taking that Jura-crust material downslope. In our foot slope, the additional colluvial layer, which has an intermediate resistivity, appears. So this is where our eroded material has been deposited. So our colluvial material is reworked debris from that slope and that Jura-crust. So it is often depleted in macronutrients and is acidic. So our passive seismic also can be broken up into our 3 vertical heterogeneous sections. So we do have our hilltop, our mid-slope, and our foot slope. However, this instrument only picked up 3 subsurface layers compared to the electrical resistivity tomography. So across all subsections, we see a thin capping material, where in the hilltop, this is of intermediate velocity, which we attribute to our Jura-crust material. And then below this cap, we see a lateritic weathering profile, which is the collective term for the pallid zone and the saprolite, which we saw in electrical resistivity tomography model. So this layer here is where our clay, salinity, or water is being stored. And then bounding all subsections is our high velocity bedrock. We do see a variation in the capping material in the mid-slope, and we see a fast velocity capping material, which is not actually there in reality. And we'll talk about why our model has picked this up later on, but that is one thing to note about this device. And then in the foot slope, that capping material has intermediate velocity again, which we attribute to our colluvial material. So looking at the results of the devices together, we see that the hilltop depicts the Jura-crust at the same depth from both instruments. However, the thickness of the lateritic weathering profile has actually been depicted deeper in the electrical resistivity tomography model across all 3 subsections compared to the passive seismic. We also see this trait with the colluvial material, and we'll explain reasons for this later on as well. We see that the hilltop has the deepest bedrock and the mid-slope has the shallowest. And this is because that deep-rooted native vegetation in the hilltop has actually stabilized the structure of the soil. So that Jura-crust material is still present because the roots are holding it together. And this also increases soil water retention, which promotes chemical weathering. So that weathering has produced that really thick lateritic weathering profile, which makes the bedrock deeper. And then in the mid-slope, the lateritic weathering profile is the thinnest because of that downslope erosion of that jury crust material and at the top of the pallid zone, which exposes it at the surface and causes deep gully networks. In our foot slope, we see that addition of the colluvial material because of that downslope transport of the pallid zone and jury crust. We also see that the shallow-rooted vegetation here also destabilizes the soil, which promotes it to more erosion downslope. However, we do still need to ground truth the model results through either a borehole, a soil pit, or trenches. However, soil pits are labour intensive and we don't want to dig that far, so we employed the use of a borehole. And while this does disturb the subsurface, it is important to cross this with our model results. So our borehole drilled down to 29 metres and it did not hit bedrock. And this supports the bedrock depth depicted in both of our geophysical instruments. The borehole also found that the water table sat at 10 metres, and at this depth the clay and the salinity also increased significantly, which corresponds very well to the top of the lateritic weathering profile and pallid zone that our electrical resistivity tomography profile depicted. So what did we find? We found that both instruments actually work best in our environment, and that is because the high salinity and clay content of that pallid zone and top of the lateritic weathering profile has actually made the electrical resistivity tomography model overestimate the depth because of it increasing its conductivity. And then we see that the passive seismic is actually ineffective in depicting layers of similar composition. And that's the case for our pallid zone and our saprolite. That is why we have put it together into a collective lateritic weathering profile. So the difference between the pallid zone and saprolite is not enough for the passive seismic to depict a velocity contrast in that layer. We also see that rock-like material in the mid-slope is a result of the poor coupling of the device with the eroded sediment of that slope, which actually increases the amount of air spaces around the spikes of the device, which increases the velocity of ambient noise. So this has depicted a rock-like material that's not actually there in reality. One other thing that is to note is that passive seismic is very sensitive to wind, noise, or footsteps, and this must be taken into account if you choose to use this in your environmental setting. So why is hill slope data important? Well, you can upscale this to landscape or catchment scales, and you can use this to simulate hydrological dynamics. So what layers are storing our water, which we saw was our pallid zone, and how quickly does water move through the profile, both laterally and horizontally. And we can predict disaster impacts, so like waterlogging and salinity. We can also use this to simulate biogeochemical dynamics, so how well our topsoils are holding our fertilizers or how quickly that moves through the profile as well. And all of this can help manage natural resources, land use planning, and overall agricultural sustainability. So we found two instruments that work very well in depicting subsurface structure. We have also presented these methods in detail across a very intensely weathered lateritic hill slope. Our next steps are to use this data to depict depth to bedrock across a catchment and depict landform, spatial distribution, and then simulate hydrological dynamics. Thank you.
Thank you. Questions, please.
Thank you, Jesse, for your good presentation. My question is, who will be the end user of your research outcome and how they will use this research outcome? Thank you.
So it's most likely going to be researchers using this data to help them with their hydrological models or their carbon cycling models, and they'll use either the sediment layering or overall depth of bedrock to do that.
Thank you for the presentation. I know you mentioned that using both the technologies in conjugation work best for the land, but if there's reasonable difference in the depths by both, which one's still more reliable, would you say?
So in our landscape, the limitation of the electrical resistivity tomography is actually supplemented by that passive seismic data. So our bedrock was actually overestimated in depth, but that passive seismic actually gives us a very accurate depth to bedrock, but we can't then only use our passive seismic because it doesn't give us that 2-layer of our lateritic weathering profile zone, so it doesn't give us our pallid zone or our saprolite, which might be important for hydrological models because that's where your water is going to be sitting. So that's why we use them together.
Yeah, that makes sense. Thank you.
Any other questions?
So, based on your thing, if I want to plant trees on the landscape, where would you suggest to plant? So that could be one of the applications. There's a lot of tree planting going on in Western Australia.
So where would I plant them?
Yeah.
In that foot slope area, probably. I wouldn't, or on that mid slope, you could probably plant them there, but it's very unstable. So.
This is what the water is coming down.
Yeah.
And then you said that the trees are doing a great job on the top, and so that where should
Put them probably in that foot slope so it can take up that additional groundwater, so then you don't have that movement of salt or clay in the subsurface. So it then doesn't change the hard pans or increase salinity risk.
Yes. Anyone else? Oh, great.
So Jesse, you said that noises like footsteps can interfere with the seismology, right? Well, we do have a seismogram at the farm, and we get 100-something plus events a year, just normal seismological events. And we also get the Boddington mine explosions as well recorded. Did they interfere with your project?
Did they impact our project? No, because we did our project at midnight, so I don't think they're blowing up anything then. Or I hope not, at least.
And a quick one, I'm not a hydrologist, so I could be way off the mark here, but I wonder, I also don't know what the profile is like as you come this way over the scarp, but I've been listening to the media around sort of all of the companies that are drawing water out of bores that may or may not be licensed and metered from our hills and Gin and so on. Does this sort of instrumentation have a role in, Coca-Cola maybe using it to understand what they're doing?
They could use it. They could use it to do, put it around boreholes and things like that, I suppose.
Thank you. Excellent. Well, please thank Jessie for her wonderful presentation. Okay. Now, our final speaker before the afternoon tea break is, I believe, Dr. Samantha, no, not yet, oh, sorry, not yet, Samantha Harvey from the School of Molecular Sciences. Samantha is a PhD researcher at the ARC Centre of Excellence in Plant Energy Biology. She's been recognised for her exceptional academic achievements and contributions to research, having received both the Sir Eric Smart Scholarship for Agricultural Research and the GRDC Postgraduate Scholarship. Samantha completed her undergraduate studies at Murdoch University, earning a degree in molecular biology and biomedical sciences, followed by an honours degree in biological sciences. During her academic career, Samantha has been actively involved in groundbreaking projects aimed at mitigating frost damage in wheat crops, in addition to Plants for Space. Currently, her research is centred on understanding how the timing of nitrogen application affects wheat protein composition. This critical work holds significant implications for agricultural practices in crop protein optimisation. Her supervisors are Professor Harvey Miller, Dr. Katarina Belt, and Dr. Hui Xiao. Today, Samantha will be presenting her findings on this important topic, highlighting the intricate relationship between nitrogen management and wheat protein quality. Please welcome Samantha.
Thank you for that introduction. First of all, I'd like to acknowledge my lab group, the Miller Laboratory. I have a few supporters here today from my lab group, and this project is also supervised by Rowan Madden. He's my JDC supervisor. The importance of my research stems from the need to optimize nitrogen utilization, to improve wheat grain quality and yield. Now, we already know that adding different amounts of nitrogen can improve quality and yield alone. However, it is unknown whether or not the timing of nitrogen application, in this case, split nitrogen applications, has an effect on wheat quality and yield. Now, why split nitrogen applications? There are some important things to consider with split nitrogen applications that can be agronomically beneficial, that being cost. Splitting nitrogen applications can essentially split the cost. That's because if we put on half at the beginning, half the amount of nitrogen at the beginning, we could then wait and see whether or not we do need to add more nitrogen later on in the season. In addition to this, there could be in-season savings on the actual cost of nitrogen itself. The environment. So if we're only putting on half of the amount of nitrogen, we do have environmental benefits in this because we have less runoff, as well as cost benefits because we have less nitrogen to lose. And then last but not least, quality. Studies in South Australia have found that split nitrogen applications can improve wheat grain overall protein percentage without compromising yield. However, we're not really sure yet if this is the case in Western Australia, so that's what I'll be looking at now. So, I'll be using the power of proteomics to discuss this critical question. And the difference here when we're looking at protein composition compared to total protein amount is when we're looking at total protein, we will put the grain into a near-infrared reader and then we'll get a total protein percentage. However, when we're looking at protein composition, we get our samples and we do some extensive sample preparation. We'll then do a protein extraction, and then we put it into a machine called a mass spectrometer, and we'll get a lot of different proteins out. So we'll get, I believe I got about 1,600 proteins per sample, and then we'll do a data analysis using MaxQuant and Studio to get these protein groups. So I put the question to you, can split nitrogen applications influence wheat grain protein composition? I'm going to take you on a journey to my field trial last year, 2023. It was in Merredin, which is a small town in the central eastern wheat belt here. My trial was at the Department of Primary Industries in Merredin, where they were able to help me with seeding and harvesting. The benefit of Merredin is it has low disease pressures, and my soil was just sandy loam soil, just to put things into context. Now, for this trial, I had 4 nitrogen amount treatments. These treatments were not split nitrogen applications. They were just based on amount of nitrogen, so we could use this to use as a control. And then we had 2 split timing treatments, so 2 nitrogen timing treatments. So my 2 time points were week 6 and week 12. So I had 3 groups essentially. I had a week 1 treatment group, a week 6 treatment group, and a week 12 treatment group. Now, the week 1 treatment group received 65 kilograms per hectare of nitrogen during seeding, and then an additional top dressing of 65 kilograms per hectare of nitrogen. When I say nitrogen, I mean urea. And then the week 6 received 65 kilograms per hectare also at week 1, and same with week 12, they also received 65 kilograms per hectare at week 1, all during seeding. And then for the week 6 treatment group, 6 weeks later, they received an additional 65 kilograms per hectare of nitrogen. And the week 12 treatment group, 12 weeks later, they received an additional 65 kilograms per hectare of nitrogen. So this was my trial here. I did have 6 commercial wheat varieties. However, I'm only going to be showing you the results today of our Scepter, which is a bred wheat. Each plot was around 1.8 metres wide and 15 metres long. So Because we had such an interesting year last year with the weather, we do need to take into consideration the average temperature and rainfall during our trial. Now, we did unfortunately have quite a dry year last year, and you can see that especially when you compare that to the historical data. Because of that, we did likely have some differences in protein composition because it was so dry. However, I put to you, there's no such thing as a perfect year. And we are likely to get something like this in future years, so it's good to know how best to tackle them. Our trial was planted mid-May, and then when we put on the 6-week top dressing of nitrogen, that was just before July, and then the 12-week was start of August, and then we did harvest this crop end of mid-December. So, how did it measure up for yield? As expected, our zero nitrogen treatment did receive little yield. However, in between 65 kilograms per hectare of urea and 260 kilograms per hectare of urea, we did have no significant difference between those three groups. Additionally, our week 1, 6 and 12 split nitrogen application treatments also had no significant difference between those groups and no significant difference between the amount of urea at all. Since we didn't have any differences in yield, we did then look at total protein percentage. Conversely, as opposed to yield, we did get differences in protein percentage according to the amount of nitrogen that we added, and that scaled up from from zero nitrogen to 260 kilograms per hectare of nitrogen. However, again, our week 1, 6 and 12 didn't have any significant difference between each other or between any of the nitrogen treatment groups. Now, this could be a good thing. It could mean that it doesn't matter when you put the nitrogen on, which there could be major benefits to that. As I said before, that means you could do the split urea applications and you're not going to get a negative impact, and you can put on nitrogen when the nitrogen price is low. So, how did protein measure up? First and foremost, I wanted to see whether or not the major functional groups of proteins actually changed according to the amount of nitrogen that I added. So, the major groups across all treatment groups for weeks 1 to 12, were storage proteins, mostly storage proteins, stress-related proteins, major carbohydrate metabolism-related proteins, protein synthesis-related, chaperone proteins, DNA synthesis-related, development-related proteins, glycolysis, or protein degradation-related proteins. And this is how the abundance of those proteins represented themselves across the three treatments. So, for week 1, as you can see here, approximately 33.59% of the proteins that I identified were storage proteins. Similarly, for week 6, approximately 3.267% of the proteins were storage proteins. And for week 12, about 35.05% of the proteins identified were storage proteins. So as you can see, there's not a huge difference between the functional groups, but that doesn't mean there's nothing happening, because there could be changes within those functional groups between proteins where some proteins will compensate for others. So then I wanted to do a differential expression analysis to find out what's actually changing. Here, as you can see, this is a volcano plot, and each of these little dots here is a different protein group. And as I said before, I got about 1,600 different protein groups. The grey colour means it either didn't reach the p-value significance of 0.05 or it didn't have more than a 20% difference. However, the coloured dots here, being the green and purple dots, mean that they were significantly different between treatment groups. So, and they had roughly about a 20% difference. Now, there's not a lot of protein differences, but some of them were significant. For example, we did have a downregulation here of high molecular weight gluten subunit protein in the week 6 compared to week 1 treatment groups. Similarly, when we look at week 12 versus week 1, we also did get differential expression. However, we got less of this alpha amylase inhibitor protein. Now, this is interesting because alpha amylase is important for starch degradation. Therefore, having more of this protein is having less inhibitor, you're going to get more starch degradation, and therefore you're going to get maybe changes in development or germination. And then in addition to this is interesting because these amylases can be used in bread making. A lot of bakers will add them to increase the likability or the taste of your bread. So, in summary, the timing of nitrogen application didn't change the yield or protein %. However, this could be a good thing. It could mean that split nitrogen applications may not negatively impact agronomy. However, this will need to be repeated in other years to make sure that this is consistent. There were minor differences observed between protein groups, but again, to back this up, we do need to do quality testing. And the quality testing that we do is we actually send the flour off to be tested to see how it works with bread making and how good it is. And then we need replicate trials. And then there were significantly different protein expression between the different groups. So, how can this data actually be leveraged? How is it useful? First of all, by knowing that protein composition can change according to the timing of nitrogen application, we can attempt to optimize for protein composition. This could bring commercial value when we're considering gluten proteins. This could bring value in terms of proteins that are important for seed storage. They could be proteins that are important that help with germination or even crop resilience. Next, we can develop a targeted protein analysis. This is exciting because if we can work out which proteins are stable according to the timing of nitrogen application that don't change, we can then develop biomarkers that can tell us how our grain was treated in the past that will help us make better nitrogen management decisions in the future. It can also be good for product development. So if we're going to actually be targeting specific proteins, we can then extract those proteins for things like fake meat, for example. And then, of course, last but not least, investing in the future. It's really important that we have a solid molecular understanding of how nitrogen impacts protein composition. This will help us better understand anything that we need for future breeding programs in terms of nitrogen. Thank you.
I jumped the gun on your doctorate, but I can see that you're well and truly on your way there.
I'm trying my best.
You've done a great job at explaining something very technical to its applied relevance, so that was excellent. Questions?
Yes, Jackie.
Not sure I need this. Sorry, Phil. Great talk, Sam. Just a couple of questions. It's a bit cheeky because you haven't actually presented it, but you mentioned you had different cultivars in your field trial. Did you see a difference between the different cultivars?
So in terms of the agronomic data, they were very, very similar. However, we haven't done the protein composition studies on them yet, but the yield and protein were very similar.
And you mentioned obviously doing replicates again this year. Are you looking at different sites as well to see the effect of different environments, or will it just still be just at Merredin?
So I did do a trial in Shenton Park, and I will be crunching that data as well.
Sorry.
It's not a field trial, but it was a bit of a tricky year this year, being the last year of my PhD.
Interesting, thanks.
Thanks.
Excellent presentation. You made it so simple
Oh, thank
for
you.
me to understand. General comment. My understanding currently, the market value is based on the total protein content of wheat. I'm wondering, do you think in the future they will look into protein composition?
Yeah, so that's the idea. I guess at the moment, the value of wheat itself isn't graded on the total protein percentage. We just have to kind of meet a market threshold at the moment. So there's an amount that you have to make for you to be able to sell your wheat. And yeah, it could be an avenue in the future to generating new products and new industry in Australia.
There's a question over here.
Very interesting talk, Sam.
Thank you.
I'm Bob French from Deeperd in Merredin.
Oh, yes.
And you're probably aware that there's been some concern expressed recently about the low protein content of Western Australian wheat. And I'm just wondering whether you think there are any implications of your work for improving the overall protein content, and what you might think those implications are.
Look, we were actually hoping that the split nitrogen application would have a similar result to South Australia, where it did increase the protein without compromising yield, but that really didn't happen, unfortunately. But that would have been very interesting if it did. I don't really have a comment on that at the moment, but I'd love to chat with you about it afterwards and see what inputs you have.
Sorry.
Thank you for a nice presentation. You mentioned that the different rates of nitrogen and timing had no effect on yield. I was interested in the screenings because obviously it was a very dry season.
Mm-hmm.
And I don't know if there was any frost or, the effect of drought on screenings from the high nitrogen levels. I wondered if you could comment on that.
Yeah, look, I haven't done the screening analysis yet. We do plan to do that. The grain, I know it, but looking at it with my eyes, it looks good. It didn't look like they had anything huge, major problems. But yes, we do plan on using the GRDC area at UWA to do the screening tests.
Thank you. Again, nice presentation. I think you did not get a response because it was a dry year. Otherwise, you would have had a split dose application. This is always a challenge for PhD students. The other one is you could look at a huge amount of CVT, cultivar variety trials across the state of Deeperd, and maybe strategically get some samples and look at the protein composition. Very easy to do it, and I'm sure Deeperd, particularly Bob and others, may be able to help. That'll be really very fascinating from Esperance all the way to Geraldton to Southern Cross, and this is quite good data you could get, and then relate to with the environment and other parameters. Thank you.
That would be great. That data would be gold.
Sorry, I think any other questions? I'm sure that Samantha will be more than happy to have a chat with you in the tea break, because I think we need to move on. So that was fantastic, and also highlighted the connection between some of the work that's going on as well, because you've sort of been talking about how this could be adaptive in the way that farmers are managing, and they can be modelling that through the sorts of tools that Michael's producing. So you guys should definitely have a chat. So please do stick around for the tea break, but I think Siddique will tell me about that.
Thank you very much for ably guiding that. Drink. Next slide. Thank you very much again for leading us through the first half of our student presenters. Please accept this as a token of appreciation, which we have done. Now the next slide. Yep. I now invite you to join the Bayliss foyer for afternoon tea, an opportunity to discuss the excellent presentation so far from 4 speakers. We will return back here at 3:20 sharp. So have a tea, coffee, come back so that we can have 3 more presentations. Then the excitement starts after that. Thank you. Welcome back. So we'll make a start now. So it's my pleasure to introduce the chair of the second session, Professor Patrick Finnegan. Patrick is the head of the School of Biological Sciences here at UWA. He has researched plant molecular physiology over his entire career. Patrick has focused on phosphorus and nitrogen nutrition in native plants that grow on our nutrient-impoverished soils. Over the last 15 years, he has been doing that. Patrick hopes that the contribution of his students and himself will one day be translated into more sustainable agricultural system. In fact, it is already happening, Patrick. A lot of those phosphorus mobilization strategy and things are being used in crops, and soil amendment strategies are being used. So that's a good one from your group and others. Since joining UWA in 2003, Patrick has also been enthusiastic teacher of the undergraduate students in biology, plant molecular physiology, and science communication. Please welcome Patrick.
Well, thank you very much for the introduction, Siddique. As you can see, I'm a bit more presentable today than I was when this photo was taken, but I really enjoy being out in the bush and enjoying the nature that we have around us. Welcome to the second session this afternoon, and I hope everybody had an enjoyable afternoon tea. Our first presenter after that break is Sarah Babington. Sarah is a final year PhD student at the UWA School of Agriculture and Environment, and her research is in collaboration with the University of Queensland and supported by the Meat and Livestock Australia. Sarah previously completed a Bachelor in Veterinary Science with honors at UQ, after which she spent several years working as an on-plant veterinarian for the Australian Government Department of Agriculture and Water Resources at various export slaughtering establishments across New South Wales and Queensland. So Sarah is very well traveled. She then moved on to work with the RSPCA Australia as their scientific officer for farm animals, where she continues to work part-time to advocate for improvements in farm animal welfare. Her PhD supervisors are Associate Professor Dominique Blanche, Professor Shane Maloney, and Dr. Alan Tilbrook. Today she'll be guiding us through identifying and validating novel biomarkers for sheep welfare. Please welcome Sarah.
Alrighty. Thank you so much for having me, everyone, today and providing me the opportunity to talk to you about the research for my PhD. So to begin, I'd just like to acknowledge those who have made my research possible. So my research would not be possible without funding from Meat and Livestock Australia, as well as my supervisory team, and those who were all involved in helping my research. So my lab group, overseas exchange students, Rob and Celeste from the UWA Molecular Biology Lab, and of course the sheep and lambs that we used for my experiments. So to set the scene for my research, I first want to define what I mean when I say animal welfare. When I talk about animal welfare in the context of my research, I've defined it as the state within an animal in which relates to what they experience, which is going to be influenced by both internal and external factors. So using this definition, we can see here that the experience of an animal is going to vary along this dynamic spectrum from both positive to negative experiences. And it's going to be the balance of these overall experiences that will actually dictate an animal's welfare over its lifetime. So when we think about how we assess animal welfare, we've got our sheep here, and typically we would apply some type of stimulus. So this might be something like a change in environment or some type of husbandry procedure. And then we would take a combination and collection of physiological indicators, behavioural indicators, and neurobiological indicators. And we would use these indicators in combination to look at how that animal is responding to the stimulus, which we would then infer what that means for their welfare. Now, while these types of indicators that we use provide us really important information about what an animal, how an animal is responding to an event, they're still somewhat limited. They can be subjective, for example, so we can look at an animal and think it is showing signs of fear, but that doesn't necessarily mean that animal is scared. They can be non-specific, so we can have hormones that might increase when an animal is stressed, but they might also increase after that animal has eaten. They can vary depending on what biological samples they're measured in. So for example, we might measure something in blood, and then we measure it again in saliva, and it has a completely different measurement. They can be challenging to interpret because many of these indicators involve multiple complex body systems. And particularly for neurobiological indicators, they can often be really impractical measuring on an on-farm setting. And these three types of indicators together mostly only look at how an animal is responding to an event rather than what that animal experiences. So this brings me to the focus of my PhD research, in which I took an alternative approach to assessing animal welfare. So here again we have our sheep, and we're going to apply that stimulus, but now imagine that we had an indicator that could link to the central processing mechanisms within the brain that were responsible for generating the real-time experience of an animal. So there are a number of indicators in human biomedical science that have been linked to changes in experience and mental state in humans, but these are yet to be explored fully in the context of animal welfare. So for my PhD research, I evaluated 10 candidate biomarkers from human biomedical science and did basically two main experiments where I induced differences in the experience of sheep. But due to my time constraints today, I'm just going to focus on my second experiment, where I specifically looked at candidate biomarkers of the experience as it relates to pain. So our first problem is actually designing an experiment where we'll induce reliable differences in the experience of lambs. So in Australia, there are a number of procedures which we perform to sheep that are considered painful, one of them being castration. And there are two main methods in which we castrate our lambs in Australia, one being surgical, and the other being rubber ring. Now, rubber ring castration is the most commonly used method because it's seen as preferable to be less painful to surgical, but it is still painful. To demonstrate the point that rubber ring castration is still painful, it's the one that I selected for my experiment. So I'm going to show you a video here, which is basically taken at the end of my experiment where the lambs that hadn't been castrated had to be castrated before they were released on farm. So let's hope it works. So here in the video, you can see that the lambs are showing behavioural signs like their heads are down, they're stamping, their tails are wagging, they're crouching, they're sitting down, and they're huddling. Now, these are all behavioural indicators that we associate with a lamb being uncomfortable and in pain. So that brings me to my actual experimental design. So I had 4 main treatment groups, and I used the rubber ring castration method. So we had our first method where I rubber ring castrated my lambs and they were provided no form of pain relief. Second, I had a group where I rubber ring castrated them but provided them a non-steroidal anti-inflammatory, which was meloxicam, and a local anaesthetic, which was bupivacaine. My third group, I sham castrated them. So this is where I pretended to castrate them but didn't release the rubber rings. And my fourth group, I sham castrated them, but they were provided the combination of pain relief. In terms of what I measured, so first up I measured cortisol, which you guys might be familiar with. Now this is a very commonly used measure of physiological stress, which has been validated in both animals and humans. And I used this to basically validate whether or not I was actually inducing a difference in at least the physiological stress response of my lambs. In terms of the candidate biomarkers that I assessed, I looked at substance P, thiol oxidation as a marker of oxidative stress, and then I looked at proteomic changes. So first up, I'm just going to briefly touch on the 3 candidate biomarkers that I looked at so that you get an understanding as to why I picked them. So we've got substance P. Now substance P is a neuropeptide and it's mediated through the neurokinin receptors system. The important thing to note here is that substance P and the neurokinin receptors together have been associated with modulating neurogenic pain and inflammation. This system has also been shown to regulate behaviour and emotional processing, so things like anxiety and depression-like states. Research in calves has shown that substance P can be elevated after painful procedures, such as castration and dehorning, but there's yet to be any studies that have actually looked at substance P in sheep. Now, thiol oxidation. So thiol oxidation specifically looks at the percentage of oxidation of thiol groups in blood, and it's used as a marker of oxidative stress. The reason oxidative stress is interesting as a biomarker of pain is because oxidative stress has been linked to stress conditions as well as neurodegenerative diseases. So we think it's obviously linked to something within the brain. Now, in my first experiment, I actually also looked at thiol oxidation, and I found that it was elevated in adult sheep 1 hour after a psychological challenge, which in the first experiment was my sheep were isolated from the other sheep. So I wanted to look at whether or not there was a difference when we applied a different type of stimulus, such as a painful one. And now I've got proteomics. Now this is kind of like a whole group in itself, but basically proteomics studies the changes in the types and expressions of proteins. And luckily we've had a few talks that have kind of explained it a bit for me, so I have an easier job. But the reason that I chose to look at it in this situation is because if we could find proteins, it could provide us insight into the really early biochemical cellular changes that are happening when an animal is experiencing pain, which we don't yet fully understand. Now, using the techniques that I used in my research, changes in proteins have already been associated with stress, inflammation, and healing in dogs after castration, as well as calves after dehorning. So that brings me to my results. So first up, we're going to look at cortisol, which, as I mentioned before, is our measure of physiological stress. So first up, we'll look at the sham castrated group, so the blue line, and you can see here, let's see, this might be good. You can see here that before and all the way after the treatment, there wasn't really any significant change in cortisol. When we look at our sham castration group that was provided pain relief, so the green line, again, from before the treatment and after, up to 96 hours after, there was no significant change in the cortisol. Now we look at the castration group that were provided pain relief, you can see there is an increase, but this wasn't actually statistically significant. So again, from before and after the treatment, there was no significant change in the cortisol. And lastly, we've got our castration group that wasn't provided pain relief. And you can see here that there was a significant increase at 30 minutes as well as 60 minutes after the treatment. So based on these results, it suggests that ring castration without pain relief induced a physiological stress response in my lambs, which suggests that the lambs were at least experiencing a difference in their stress response, which suggested to me that they likely also were having a difference in their experience. So that brings me to the candidate biomarkers that I looked at. So substance P. Now I looked at this before as well as 1 hour after. The reason I looked at it 1 hour after my treatment was because substance P has been linked to increasing just after cortisol. And as you just saw, I saw cortisol to initially increase significantly at 30 minutes in my ring castration group. So before I performed any treatments on my lambs, there was no significant difference between substance P. And when we look at 1 hour after the treatment, we can see not only was there no change, but there was no significant difference between the treatment groups that I assessed. Next up, we've got thiol oxidation. Now, thiol oxidation I looked at before, 1 hour after, and 48 hours after. The reasons that I picked these times is because my first PhD experiment, I saw that significant increase in the thiol oxidation at 1 hour after. And studies in humans and animals have shown that levels of oxidative stress can remain elevated after a stress up to 48 hours after. So when we look at our measures before, you can see that there was no significant difference between my treatment groups. At 1 hour after, not only was there no significant change, but there was also no significant difference between the treatment groups. And there was a similar story at 48 hours after, no significant change and no significant difference between my treatment groups. Lastly, we have our proteomic results. Now, due to resource constraints, I was only able to look at the protein changes between my group that was castrated and the group that was castrated with pain relief. So this is a similar graph to the one that you saw in a previous presentation, whereby those little black dots at the bottom there, these were the proteins that I quantified. In my scenario, we only quantified 287 proteins. And this red line, anything above that would suggest that there was a significant change in the expression of proteins between the groups. So you can see here that before the treatment, there was no significant difference in the expression of proteins. When we look at 30 minutes after the treatment, all of those little black lines, you can see none are above the red line, which suggests there was no significant difference in the expression of the proteins that I quantified. 2 hours after the treatment, very similar story, no significant difference in the proteins that were quantified. And I measured protein changes up to 96 hours after, and I can say that it was a very similar story in that there was no significant differences in the expression of the proteins that I quantified up to 96 hours between the groups. So what do these results mean? To summarize, I saw a significant difference in cortisol, in which the group that I ring castrated without pain relief had a significant increase at 30 and 60 minutes after, whereas the other groups did not. But when we looked at our candidate biomarkers of pain, I saw no significant changes in substance P, thiol oxidation, or the plasma proteins. Now, I'm not going to lie, these results were a little disappointing at first and unexpected, but my supervisors continued to remind me that there's no such thing as no result. There are just results that we need to interpret. So, ring castration causes an ischemic injury. So this would be the same as if we were to put a rubber band around your finger. When you first put that rubber band around your finger, you're going to get a lack of oxygen to the tissues. If you were to leave that rubber band on, you're going to begin to get cellular damage and acidosis. And if we were to go beyond this point, eventually we would begin to get tissue necrosis. Now, it's the cellular acidosis that actually activates afferent nerve fibers in your finger and sends a message up your spinal cord to your brain that you perceive as pain, which would hopefully mean you took that rubber band off your finger. Now, when we apply this to the rubber ring castration method, we can see here that we have nerves that will run all the way up from the testes into the brain of the sheep. Now, my results so far suggest that there is likely little to no systemic inflammation involved when we rubber ring castrate lambs. Instead, we cause an ischemic injury. So when we apply the rubber band to the testes, over time we get that cellular acidosis and the tissue necrosis, which activates our afferent nerve fibers, sending that pain signal up the spinal cord to the brain, where the lamb then perceives it as pain. So in terms of research implications, my results so far suggest that ring castration is associated with very little inflammatory pain. Instead, most of that pain is likely neuropathic. The tested biomarkers that I looked at not only have been associated with pain, but they've also been associated with a lot of inflammatory states. So perhaps they're better for assessing inflammatory types of pain, such as the pain that would be caused from a surgical castration. It also raised a number of really interesting factors that we need to consider for future research when we think about finding biomarkers of pain. So we'll need to think of things like age, whether or not the animal is young or old. For example, I didn't see a significant difference in my thiol oxidation in my young lambs, potentially because they have a higher antioxidants capacity, whereas I did see a significant difference in my adult sheep after they were just psychologically stressed. We need to think about the time that we're measuring these biomarkers. For example, we not only need to measure before the stimulus, but when exactly after are we going to measure? Because that will likely depend on the biomarker that we're wanting to target. And we need to think really carefully about the type of pain that we're trying to measure. Is it an inflammatory or a neuropathic type of pain? My research also raises some really interesting questions about the pain relief options that we currently use. So currently for rubber ring castration, we mostly use a mix or by itself a non-steroidal anti-inflammatory or a local anaesthetic. Now, research so far has shown questionable results in terms of the efficiency of these pain relief options for rubber ring castration. One of those reasons might be because these two agents mostly look and address pain by reducing inflammation, which my results suggest there is little of when it comes to rubber ring castration. So perhaps we need to explore alternatives that are more focused towards addressing neuropathic types of pain. So while you can see that there is still lots of work to be done, and I still have a lot of data analysis that I'm digging through, I just want to leave you with, I guess, a sum up of what I'm hoping my PhD research will contribute to, which is changing the way in which we assess animal welfare. So again, we have our sheep, and we're going to apply that stimulus. And hopefully, in the future, with the help of my research, we will have these indicators of experience that we will then also be able to look at physiological, behavioural and neurobiological indicators, so that when we look and assess an animal's welfare, we have a more complete picture of their overall welfare. Thank you.
Thank you very much, Sarah. We have some time for questions. Yes, please.
That was a very interesting talk, Sarah. Bob French from DPIRD. You started the talk by justifying the work in saying that we need markers of welfare in animals that are more easy to apply at a, say, in a real-world situation. I'm struggling to see how the biomarkers that you were looking at, you might apply them on a farm or in an animal house or a context like that, and I wonder if you might comment.
Yeah, really good question, because obviously that's kind of one of the priorities is that if we are going to have measures, they need to be applicable on farm. So the measures that I used were all in blood samples, which obviously aren't necessarily super applicable right now to a real life setting. But what we're hoping is that we will first validate them in blood or say tissue, and then we'll be able to transfer that to less invasive measures. So we've already got work ongoing at looking at saliva samples, which would at least be a little more practical in terms of like an on-farm setting. Another thing that these could be really useful for is if we had biomarkers of experience, it might actually help us validate some of the really easy markers that we already use, like behavioural indicators. So we could actually validate whether or not the behaviour that we're seeing is actually causing those differences in experience that we think they're causing, which could then also allow behavioural indicators to be better validated and used on field.
Yeah, thanks for the very nice illustrative presentation. Someone commented during coffee break whether Rosanna is paying me for asking questions. Okay? The reason I'm asking question is students feel very happy. At least someone is seriously listening to them. And also, secondly, the subject is very interesting, so I'm very curious. My question is in methodology. I'm curious why you chosen a methodology before and after instead of a control group, without castration of the same age group. Because in sheep you can get twins. So you can use a control group and see what's happening to all these biomarker and castrated group. What is the reason for choosing this kind of methodology? I'm curious.
No, really
I'm
good
not saying this methodology is wrong, but I'm curious why you are using before and after instead of control. For example, we are doing a lot of work on forest fire. So we try to look at a control group without fire and with fire, not before and after. So I'm just curious.
Great question. That might be a fault of mine by not explaining that properly. So we did have a control group. So my sham castrated group, they were just handled, but they were not castrated. And then I also had another control group where they weren't castrated, but they were provided the pain relief, because I specifically wanted to look at, exactly as you pointed out, not only the groups that were castrated with or without pain relief, but groups that weren't castrated at all with and without pain relief. Yeah.
Time for one more question. Graham.
So, Sarah, I think the impact of your work is actually more enormous than you were telling us, because it seems to me that we've been giving advice to farmers about pain relief, making them feel good about themselves as they cut off, or as they put the ring on, but in actual fact, it's all been wrong. It's all been the wrong medication. So, for me, your work is really important in showing that maybe we need to reassess that in complete situation with respect to how we handle castration.
Yeah, it's something that I'm very passionate about, particularly because there's a lot of work at the moment in regards to mandating pain relief for castration procedures and stuff for sheep. And although this is a really positive step, because it's obviously pushing farmers towards recognising and working towards using pain relief, there isn't much point if the pain relief isn't useful. So I think the first step is actually to reassessing the pain relief we're using, because it's clearly not as efficient as we want it to be, and actually first off find pain relief that is efficient and then look at mandating it after that, so that farmers can actually feel good about what they're doing when they're using the pain relief, for sure.
Okay. Thank you very much, Sarah. I have a certificate for you. And a small certificate. Thank you very much. Okay. Our next speaker is Sharmin Sultana, who is a final year PhD student in the Division of Pharmacy at the UWA School of Allied Health. The title of her PhD project is Phytochemical Analysis and Bioactivity Profiling of Clover Honeys. She completed her Bachelor of Pharmacy with honors and Master of Pharmacy from Pharmacy Discipline of Konla University in Bangladesh. Sharmin started her career as a quality control officer in Septa Pharmaceuticals Limited, Bangladesh. She was responsible for the analysis of various dosage forms, assay of finished products, and determination of impurities in raw materials. She moved to Perth in December 2019 and worked as a research assistant in Be Pain-Free clinic trial of the anesthesia research team at Perth Children's Hospital from January to November in 2020. Her PhD supervisors are Associate Professor Cornelia Roker, Professor Lee Long Lim, and Dr. Kevin Foster. Today she'll be exploring identification and quantification of isoflavones in monofloral clover honeys. Please welcome Carmen.
Hello everyone, thank you for giving me this opportunity. I am also thankful to all of my supervisors as well as my advisory panel members for their amazing supervision throughout this journey. Apart from this, I am also appreciative to all of my lab mates for their cooperation. Sorry, the slides are changing automatically. Anyway, perhaps you are already aware that honey is a flavorsome natural product with high nutritional and medicinal value. There are different types of honey, such as Jara honey, Manuka honey, and so on. However, my PhD work was focused on clover honey. The reason behind this is the phytochemical composition of various honeys and their associated bioactivities. Surprisingly, there is no comparative data on the concentration of flavonoid or more specifically isoflavonoids in clover honeys. Although isoflavonoids have been identified as a main group of bioactive compound in red clover plant. So my PhD project was designed to identify and also to quantify this unique bioactive compound, phytoestrogenic isoflavonoids, in order to also to investigate other bioactivities such as antioxidant activity to tap into new opportunities that could have potential benefits for both pharmaceutical and apiculture industries. I'm not sure that why the slides are changing automatically, sorry about that. Okay, the term phytoestrogen is connected to estrogen. While estrogen is a hormone produced by human, phytoestrogen is a plant-derived compound which is known to have some estrogenic activity. On the other hand, isoflavones are found in many legumes, including clover, some of which are known to be phytoestrogens. Here you can see that the isoflavones share structural similarities with 17-beta estradiol, which is the primary form of estrogen. Here it's important to note that estrogen is primarily a hormone that is used by female for their menstruation, fertility and pregnancy. But the level of estrogen can fluctuate throughout the menstrual cycle and decrease after menopause. But isoflavone can help to regulate them by acting as agonist on estrogen receptor, which in turn used to mitigate the negative outcomes of decreased estrogen levels, such as sleeping disorder, weight increase, mood changes, vaginal dryness, etc. And according to the literature, isoflavones are the main group of bioactive compound in the red clover plant. So clover is a leguminous herb with trifoliate leaves and dense flower heads. And clover honey is a type of honey made by honeybees collecting nectar from clover flowers. When this honey is produced by placing the nucleus into a controlled growing environment that is known as monofloral clover honey. So the raw material for honey production is the flower nectar. Bees gather nectar from flower and keep it into their honey sac, where it converts the nectar sucrose into glucose and fructose. Once the honey sac is full, bees return to their hive and pass this nectar mouth to mouth onto other bees. During this process, additional enzymes are added to the enzymatically processed nectar and deposit into honeycomb, which is known as unripe honey. This unripe honey is then turned to mature honey by the evaporation of its water content. And the whole process is completed when the bees cap the honeycomb with wax to store the mature honey as a future food source. And the production of monofloral clover honey was initiated with the seedlings of clover cultivars, which were then moved into their designated plot inside shade house enclosure at UWA Centre Park Field Station, followed by the installation of nucleus. This project involved the cultivation of 2 red clover cultivars such as NSE and NFE, which were specifically bred to have their high isoflavone content, and also 2 other cultivars such as Othello and Electra. Different parts of clovers were collected alongside clover honeys to capture their respective isoflavone profile, such as leaves, flower brackets, which refers to the structure that cradle individual flower within a larger inflorescence. Also, flower nectar, unripe honey and mature honey, which were then analyzed using the high performance thin layer chromatographic technique. High performance thin layer chromatography, or HPTLC for short, is a semi-automated and advanced type of TLC. This equipment consists of 4 different parts, such as applicator for the sample, development chamber, which uses a suitable solvent mixture to separate the compounds present in a sample into different bands. Then a visualizer where these bands can be seen under different light conditions, and also a TLC scanner which measures the UV and fluorescence absorbance of this band. This is a representative HPTLC generated chromatogram, and the whole machine is operated with the Kamak VisionCat software. 8 distinct types of isoflavones were detected and measured in different parts of clovers. From the literature, a biochemical correlation between these isoflavones were identified. For example, genistein is produced from the demethylation of its precursor biocanin A. However, this genistein can also be present as its glucoconjugate genistein. Conversely, sisotrin is the glucoconjugate of biocanin A. Through enzymatic hydrolysis, both of these glucosidic isoflavones can be converted into their corresponding non-glucosidic isoflavones by releasing their glucose unit. Here you can see that I am using different color codes now and onwards to distinguish between various forms of isoflavones so that everyone can easily identify the different form of isoflavones present in different parts of clover. Similarly, diargine is produced from the demethylation of its precursor formononetin. But this diargine can also be present as its glucoconjugate diargine. On the other hand, ononin is the glucoconjugate of formononetin. With enzymatic hydrolysis, both of these glucosidic isoflavones can be transformed into their corresponding non-glucosidic isoflavones by releasing their glucose unit. This biochemical conversion allow the classification of these different form of isoflavones into 2 distinct groups. Firstly, glucosidic isoflavones such as sisotrin, genistein, ononin and diargine. Secondly, non-glucosidic isoflavones such as biocanin A and formononetin, which are the methylated form of non-glucosidic phytoestrogenic compound. Then genistein and diargine, which are the demethylated form of non-glucosidic phytoestrogenic compound. the biochemical correlation also indicates that the demethylated isoflavones are the more potent form of isoflavones between these 8 phytoestrogenic compounds. This slide represents the identified and quantified isoflavones in different plant parts such as leaves and flower brackets. Here you can see that the non-glucosidic isoflavones were predominant in both plant parts compared to the glucosidic form. Also, demethylated isoflavones were exclusively identified in the leaf extract of both red clover cultivars, NSC and NFE. This slide displays the quantified isoflavones in the clover flower nectar, unripe honey and mature honey, while a range of isoflavones such as glucosidic, methylated or demethylated form of isoflavones were detected in the corresponding flower nectar. Only the demethylated isoflavones were identified in clover-derived, unripe and mature honey. Before discussing the main findings of this study, I want to remind you that bees are the true chemists that use flower nectar to produce honey. And the result of this study illuminate a complex biochemical conversion by showing a fascinating interaction between flower nectar, honeybees and isoflavones. For example, genistein and diargine were identified in NSC unripe honey that are the demethylated form of isoflavones, while a range of isoflavones such as the glucosidic form genistein, methylated form biocanin A and demethylated form genistein were detected in the NSC flower nectar. Here it's important to note once more that bees use enzyme to produce the unripe honey, and the result of this study may indicate that isoflavones are chemically converting during the bees' conversion of flower nectar into unripe honey. The analysis also revealed an intriguing finding such as the phytoestrogenic compound present in the unripe honey and mature honey were identical, despite their variation in concentration. This is a quantitative example of the level of isoflavones in NSC unripe honey and NSC mature honey, where genistein and diargine present in the NSC unripe honey were found to increase by approximately 40 to 50% in NSC mature honey. And the result of this study indicates that there is no additional chemical change rather than the evaporation of water when the unripe honey settles into honeycomb and matures. Additionally, the quantitative data of this study indicates that isoflavone, which is abundant in the clover honey, that isoflavone or the related form of that isoflavone is also the predominant isoflavone in corresponding flower nectar and plant parts. For example, NSC mature honey has a higher concentration of genistein compared to diargine. Interestingly, genistein and its related form of isoflavones were also dominant in NSC flower nectar and plant part compared to diargine and its related form of isoflavones present. A similar pattern of result were also identified in different parts of NFE, Othello and Electro cultivars. But because of the interest of time, I have explained one of the cultivars just as an example. So from this qualitative and quantitative data, it's reasonable to conclude that both of the red clovers are potential in terms of the amount of phytoestrogenic compound. However, NSC has a higher concentration of biocanin A, whereas NFE has a higher concentration of formononetine. Secondly, a range of isoflavones were detected in the various parts of clover. However, demethylated isoflavones were the only identified isoflavones in all tested monofloral clover honey. And maybe you can remember that the demethylated form of isoflavones are the more potent form of isoflavones between these 8 phytoestrogenic compounds. Also, the level of isoflavones in the clover honey were lower compared to those detected in its plant part. However, there could still be an opportunity to develop the honey product into health food or dietary supplements. Besides, this study shed light on intricate biochemical conversion by demonstrating a captivating interplay between flower nectar and honey, where honeybees might play a vital role. That's all for the time being, and thank you everyone. Sorry for this slide.
Thank you very much, Charmin. We have time for some questions. Are there any questions? I was interested to know, okay. So while the microphone's going back there, I was interested to know whether the demethylated form of the isoflavones was being converted during the honey maturation process by an enzyme or whether it was autocatalytic.
When bees collect nectar, they actually perform various enzymatic action. Like they use invertase enzyme to break the nectar sucrose into glucose and fructose. Another enzyme like catalase that breaks, like amylase that actually breaks the amylose into glucose. Also glucose oxidase that breaks down the glucose and stabilizes the pH of the honey. So from the literature, I found that all of this enzymatic action is performed from the bee's salivary gland. So we are also assuming that the conversion of isoflavones when the bees converting the flower nectar into unripe or ripe honey is also related to the enzymatic action.
That's a very good point. The reason I asked was that in Manuka honey, the bioactive ingredient that gives that honey its premium price is methylglyoxal. And methylglyoxal is converted from dihydroxyacetone during the honey maturation process autocatalytically. So there's no bee interaction that's necessary for that conversion. But your system seems to be potentially different from that. That's very interesting.
Thank you, Charmin. She went to same school for undergraduate with me. Oh, so very proud of you. And also the similarities of biochemists to present number in the form of diagram is very unique. And the painful days I remember studying biochemistry in undergrad, like all those structure and everything, but you did very well. So it's not a question, it's just for fun. Which honey we should choose from which cultivar?
You can see that I have selected 4 different cultivars. So 2 of them is red clover and other 2. So the reason behind this, that from the literature I found that red clovers is potential in terms of the amount of phytoestrogenic compound. So that's why we chose these popular cultivars and besides 2 other less popular cultivars. So, and from the study we found that the amount of phytoestrogen in the different cultivars, the red clovers is little bit higher. But all the cultivars, they contain the demethylated form and this is the more potent form of phytoestrogenic. But in a sentence, I will say it is red clover.
Another question over here?
Thank you. Ben Biddle from DPERD. Great presentation and a bit of a methods question and then also an opportunity for other interactions. That with your experimental design, how did you control to make sure you had the same? Because obviously your bees are confined to each different clover variety in each different polytunnel or screen tunnel, how did you make sure that the bees that you put in there were the same? Which is obviously the first question. And the second question is, obviously there's varietal differences in the clover in terms of the isoflavones they produce, but is there also differences in bees from different environments in terms of the enzymatic contributions they take for the conversion of those isoflavones into the different forms and the chemical forms that come out of that?
To confirm the production of monofloral clover honey, as I mentioned that we planted this species in a controlled growing environment. So that was completely enclosed. So we are pretty much sure, or this is the way actually to produce the monofloral honey. So, and the second question.
Yeah, so that, but each beehive that you put into each tunnel, how did you make sure that those bees were all the same, that they had the same enzymatic activity? And is there is there not any differences between the bees' ability to convert?
There can be. It depends on the age of the bee or other thing, but we make sure that the bees we are setting over there, they all are pretty much with the same ages, and yeah, that's it.
Thank you. Okay. Any other questions for Charmaine? If not, thank you very much. Oh, we have one more.
Bob French from DPERD. I was just wondering, with the concentrations of the phytoestrogens that you measured in the honey, how much honey would you need to eat to get a physiological effect?
Well, that's a very good question. I was expecting that. Okay, I know. In terms of the amount of phytoestrogenic compound in clover honey, that's really less. But there are different types of method to do the hormone replacement therapy, like the bioidentical hormone replacement therapy, where the hormones can be given by pills, patches, or any other medicinal formula. And the another type is the traditional natural hormone replacement therapy, which involves the consumption of supplements or plants or any plant-derived product. So where red clover or red clover tea, they are already using as a hormone replacement therapy. And I will say clover honey will be a good addition over there, like adding a spoon of clover honey into clover tea or spread clover honey on the bread.
Thank you very much, Charmaine.
Okay.
Charmaine, a certificate and a gift certificate to thank you for your presentation today.
No worries, thank you. I need to give you this.
Our final presentation today is presenter today is Felipe Castro from the UWA School of Agriculture and Environment. Felipe obtained a bachelor's in agronomy from the Pontificia Universidad Católica de Chile, something like that, in 2014, where he developed a liking for plant breeding and statistics. After graduating, he joined the sweet cherry breeding program of that university as program manager, staying there until July 2018, when he came to Perth to study a Master of Agricultural Science, concentrating in genetics and breeding at UWA. And eventually he commenced his PhD studies in 2021. During his studies at UWA, Felipe has worked in developing statistical models to analyze breeding data and achieve higher accuracy of prediction of genetic values in early generations of self-pollinating crops, and incorporating different approaches to increase the rate of genetic gains and manage population diversity in the long term. His supervisors are Professor Wallace Cowling and Hackett Professor Kadambot Siddique and Dr. Li. The title of his talk is Sustainable and Rapid Genetic Gain with Genomic Selection in Early Generations of Field Pea Breeding. Thank you, Felipe.
Thank you. Thank you. Hello, everyone. Thank you for the introduction and for the opportunity to be here. Let's see if this works now. But I'm really excited to tell you about my project and what I've been working on the last few years. But before that, I would like to thank my PhD supervisors and all the people, companies, and scholarships that have made this project possible. With my project, I want to be able to improve how we do breeding for self-pollinating crops. And by that, I mean that I want to be able to achieve faster genetic gains, and I want to be able to sustain those gains for a long term, for a long time. So with that in mind, the objectives of my project are I want to reduce the length of the breeding cycle. I want to increase the accuracy of the genetic predictions that we do with genomic selection. And lastly, I want to be able to simulate the long-term effects of using a short cycle. This is important because we need innovations in breeding. We know that the population is growing, and therefore the demand for food is growing. When we look at research, we see that most publications agree that the increase will be over 50% by 2050, which means that the rate of demand is growing at more than 2% per year. And so we need more food. And we are just not able to grow more crops. We don't have more land available. And we need different solutions for that. One of them is breeding and getting varieties that will have more yield. How are we doing on that? When we look in the literature for genetic gain for yield, we'll see figures like this. When we see release material across the years, this is an example of durum wheat in Canada. And we see that here they achieve 0.63% of genetic gain per year. And that's the reality for most crops. Most crops are around 1% per year of gain, but we have a demand growing around 2% per year. So that's not enough, and we need to double this rate. So as I said, I work on self-pollinating crops. I work with field pea, as you can see there. And what's particular about self-pollinating crops is that we want to get a self-pollinated plant, which we want pure lines. The conventional breeding for self-pollinated crops may look like this. This is a really simplified diagram, but it tells the story. And it is that we cross the parents, and then we do a series of trials where we can be doing some testing, some seed bulking, before we take our next breeding action. And every time we do that, we are essentially self-pollinating the plants. So it may take several years of selfing before we can do the next cross. In my diagram, it will be something like that. And that easily can be 8, 10, or 12 years before we do the next cross. So there are methods that can make this faster, so take less years, but the essential selfing structure remains largely the same. My question here is, can we speed this process in a different way? Can we short the cycle? This idea comes from the breeders equation, which is a conceptual equation for breeders that relates that big R there, that's the response to selection in the next cycle, that's my genetic gain, to a bunch of variables. But today, I want to focus on the variable on the bottom. That big L is the length of the breeding cycle. And by looking at that equation, we could think that, in theory, if we make the L smaller, we should get more genetic gains. So what is this question in my breeding context? It means asking, could I cross before doing all that selfing? That will be similar to, for example, what animal breeders do. And this is a gross oversimplification of animal breeding, but it could look something like that, where candidate selection and crosses happen more dynamically and quickly than on plant breeding. So on my crop breeding, could I do something like, say, stopping there, the selfing, bring the cross forward, and then repeat this strategy? This is known as early generation selection, and it's one of the goals of my project. I want to be able to do this. This is not a new idea. It's been tried before, and it comes with some challenges, because now I don't have that selfing, I don't have all that replication. I have less accuracy on my breeding values. Those are the genetic values that I use for selection and also to measure my genetic gain. And with that lower accuracy, I have a difficult time selecting when I have multiple traits that are complex and often have low heritability. That means that of the variability I see on the field, just a little proportion, it's due to genetics that I can use when doing breeding. So my research question here is, can I make that work if I'm able to get more accuracy of the breeding values? So this brings me to my field pea population. I've been working with this population for many years now, since my master's actually in 2019. I've done a series of trials and crosses in the glasshouse. And while working on this, I focus on two aspects of breeding: how I analyze the data and how I select the candidates. The how I analyze, for me, means how I build a good statistical model to analyze data, with the idea that if I have access to more information, I should be able to get more accuracy in my predictions. So I'm seeking this optimized genomic selection model to get more accuracy. On the selection, it means that I need to decide which plants should be parents in the next cycle. To do that, I use something known as optimal contribution selection, OCS, which is an algorithm that not only tells me which plants should be crossed, but also how they should be crossed, so which couples should happen there. And it does this by trying to promote genetic gains and manage population diversity. There are some challenges here, of course, and they come with balancing. I need to balance, in the case of the model, how complex I build the model to get useful gains, and because an overly complex model can give me little yield and little benefits and maybe run over several days, so it's not useful. And on the side of selection, it means how much genetic gain can I ask for in the next cycle without running over my genetic diversity. So I tested several traits in my population, but for today's presentation, I want to focus on one of them, which is compressed stem thickness, and it's a measure that we use to measure lodging resistance. This trait is interesting because there hasn't been any real improvement on field P in the last few years, decades even, and it's one of these low heritability traits. So it's a good subject to see if this methodology works. So we know lodging and we don't like it. The stems in Ps are weak, are hollow, so when they are heavy, they tend to fall over. Then you go to harvest and you could see something like that, which is quite annoying because you will harvest soil, you will lose yield, you have contamination. We don't want that. So with this trait, we are focusing on the stems and we try to look at that area, at that section. And we want to make that stronger. To do that, after harvest, we got the dry stems and we put the dry stems on a force gauge machine there, and we compress with a specific load, and we measure how much we can compress the stem. So it looks like that, and hopefully displays. Yep. So we're compressing the stem with a specific load, and we measure the displacement that we get there. So a stem that compresses less is a stronger stem, and we want to promote that. So let's look at some results for this trait when we do early generation selection. I will focus on 3 aspects of the results. First, if we are able to see an increase in the heritability of the trait with better models. Second, if using those models, am I able to get more accuracy in my genetic predictions? And lastly, but the most important one, is am I getting an improvement in breeding values? So that's, do I get genetic gains with this? So let's look at the heritability of the trait. I will compare 2 models here. I'm comparing a model built with pedigree information and a model built with genomic information. And that's the data I have from my years of trials. And we can see that in this case, using a genomic selection model gets a higher heritability, meaning that this model is able to capture more genetic variability and in theory should be a better model to do my breeding. So it seems to be good on that side. Can I get accurate predictions with that? So let's look at the second set of results. For that, I will focus on that trial over there last year on Shenton Park. We measure compressive thickness for all those plants, and then I propose different models, and I see if the predictions are accurate or not. So let's look at the accuracy of those predicted breeding values. I will compare 4 models in this case. The first one is what I call the base pedigree model, which is essentially, it's a really simple model. I just get the observations, and I relate the genotypes with pedigree information, but I don't do anything fancy with the model. We have a base genomic model, which in a similar way is just observations plus this genetic relationship information. But then I have a set of optimized models with both genomic information and pedigree information. And when I say optimized, these are more complex models. Here I consider multivariate analysis, some correlation between residuals, some block effects, maybe site effects, spatial analysis. So it's a much more complex model. It takes more time to run. But as we can see here, it also gets more accuracy in the prediction of the breeding values. In this case, both the pedigree and genomic model get more accuracy. So that's good. It seems to me that I can get more accuracy when I optimize the model. Is that useful for selection now? That's the next stage. Can I get genetic gains with that? For that, let's focus on those two trials. So the same trial that we saw before, and this other trial, which are the trials where I got my genomic information and I trained the model. Let's look at the breeding values to get an idea of the genetic gain there. So when we look at that, we see a couple of things. From 2021 to 2023, for that same trait, that's the compressed thickness, I got an increase in the breeding values, meaning that I got genetic gain for the trait. The other thing that we can see here is that on 2023, I have an explosion on the range of the values, and that's a feature of using optimal contribution selection. I managed to increase the variability on the population. So we have gains from 2021 to 2023. How much? Actually, in this model, I achieved 13% per year of gain, which is quite a lot, but may happen on a first cycle of selection. It's not impossible to have it. And the second thing that we can see here is that the genomic model achieves more genetic gain than the pedigree model. So it seems to support the idea that genomic selection, it's better to get more genetic gains. However, this is just 1-2 years of breeding, just one cycle of difference. It's not enough time to be able to say that this will work in the future. So with that in mind, my next question is this going to be sustained in the long term? That's impossible to answer with real data in the span of a PhD. So the only thing that we can do is simulate that. I can run a breeding program simulation and see the long-term effect of taking different breeding decisions. So that's what I did. Earlier this year, thanks to the My Carroll Traveling Fellowship, I got to travel to Edinburgh, Scotland, to learn how to run breeding program simulations in the Ronslin Institute. And what we do there is that basically we model a population, and we subject that population to different strategies for analysis and selection, and we repeat that many, many times, and we get an idea of the average outcome of those breeding decisions. And this is quite useful to see trends of what could happen in the future. So let's have a look at that. For today, I ran the simulation for compressed thickness for 20 years. And well, you will see a bunch of lines here because we have, as I said, I ran this for many repetitions, but let's focus on the bold lines. That's the average outcome for every scenario of my simulation for 20 years of breeding. And as you can see on the scenarios, I have a mix of different breeding strategies. I'm using a combination of 4-year selection cycles, 1-year selection cycles, phenotypic selection, pedigree selection, genomic selection, with and without OCS, and we see that there's quite some differences between them. Let's focus on the two extreme results here. So let me get rid of the other ones. And I have the two extreme cases. The baseline there, the red one, is what I would call my base scenario, even my conventional scenario. It's what I did 4-year selection cycles just based on phenotypic selection. And the other line on top of that, it's my 1-year cycle with genomic selection and optimal contribution selection. And we can see that there is quite a big difference of that after 20 years. In fact, the conventional scenario, pretty much in line with the literature, achieved a 1.2% of genetic gain per year, while my short cycle genomic selection got almost 4% of genetic gain per year. So that means that the simulation says that this more than triples the genetic gain of the conventional approach. So, to conclude, we can see that we are able to increase the accuracy of the breeding values if we optimize the models, and that happens with both pedigree and genomic information. When we have this high accuracy, we can undertake shorter cycles, as short as 1 year here, but maybe that's a bit overkill. We can try 2 or 3 year selection cycles, but definitely less than the conventional approach. And lastly, genomic selection achieves more genetic gain, and so it seems to be a better model to promote gains in the future. Regarding the simulation, we can see that it supports that we can do selection without all that selfing, and that the gains should be sustained in the long term. So to finish off, I would like to just show that diagram again and say that if we make systematic use of all the tools that we have available now, we should be able to achieve faster and sustainable genetic gains. Thank you.
Thank you, Felipe. Any questions for Felipe?
Felipe, the animal scientists, the cleverer we've got with our accuracies of EBVs, the more exposed we've been with negative genetic correlations to other traits. So it's just a cautionary tale more than anything else, is that it's such a powerful thing to incorporate genomic information into
Definitely.
that EBV. And so I guess my question is, do you know the genetic correlations with the other traits that make you money? And
yes.
I assume that you are keeping your eye on those as well.
Yes, so there are definitely correlations, and often those are unfavorable. So it's difficult, but they are not one, which means that if you have good accuracy and you develop a strategy, you can make progress for both traits, maybe at a slower rate, but it could happen. I have a big case of that with one of my traits is black spot resistance, so that's resistant to ascochytal light. And I also have flowering time, which it makes sense that if you want shorter flowering time because you want earlier varieties, but when you have that, you expose them to more disease. So you have a negative correlation there that you don't want. But we've seen that it's possible to breed for disease resistance and at least hold that flowering time at some level, and we can even push for that a little bit, not as faster as we would want, but yeah, you need something that in mind, and that's why a complex model with multivariate analysis is quite useful.
There's a question over here.
Thanks, Felipe. Great talk and quite interesting philosophising around this. One of the other ways a lot of our breeders use genomic selection is essentially restricting their population, making their selections earlier in their selection cycle, and increasing their population sizes. Have you done any of the like a similar analysis with your populations as well in that space?
Yes, so an example I can do with that, an extreme case would be what I achieved here, for example. And I could use just the genetic data of those two trials, but you use those observations to try to predict the breeding values. It's not as good as having the actual phenotypic data of that, but I still get 0.6 correlation between a predicted breeding value and from 2021 with a predicted breeding value from 2023. So that's definitely a use that we can make, and optimizing training sets size. Yeah, that can be done. In this case, I just show what happens if you have all the information available. But yeah, definitely.
Thank you.
Any other questions for Felipe? If not, thank you very much, Felipe.
Thank you.
Thank
you.
For your appreciation.
Thank you.
And I'd like to thank all of the speakers this afternoon before handing over to Siddique for some closing comments. So, very nice talk.
Well, please join me in thanking Patrick for chairing that session, Abbie. Thank you. Sorry. So first of all, I would like to congratulate all the presenters. I think you will all agree that our PhD students did a fantastic job introducing and explaining their complex research projects in a very interesting and also an entertaining way with all the graphical images and so on. So just to reflect back, some of us did the PhD time. We didn't have all that. We had probably the overhead projectors and so on, but they have done extremely well. The other point is that all these projects have got a research question related to the industry. So those people who are not related with the university and PhD projects, this is not an isolated incident. Everywhere they are given a topic, and that topic has got some background. So for example, the last one, the pea breeding, people like Tanveer and others have been working over the years. It's a very complex one. And it also involves the futuristic outlook. Look at the modeling they have done, so that it's not just about the 3 years. But also it's a very complex process. Now the university is insisting we must finish this PhD student by 3.5 years. And it will be extremely hard, a complex topic like this and other topics you heard. So they have done extremely well. They worked very hard under the able guidance of Emeritus Professor Graham Martin, and we certainly thank you, Graham, over the last 18 years you have been helping the students. So please thank Graham for his work. So the science communication is again part of this whole thing. You know, the students were under tremendous pressure when they were selected, then they were trained themselves, and supervisors also had input. But I'm sure that this will go a long way in their career as well. Now, we have some few things I have to announce. Before we finish, I wish to share some of our upcoming events, and the QR codes are on the screen. So the first one is our annual industry forum, and the topic is, can agriculture achieve net zero? This is a very topical topic at the moment, and we have got a number of people speaking, including the Minister for Energy, Environment and Climate Action, Honorable Reese Whitby, MLA, is going to officially open. Then we have CEO of CRC for High Performance Soils, Dr. Michael Crawford, based in Eastern States. Then Rose Kingwell, our own Rose Kingwell, and also the president of the Pastoralists and Graziers Association, Tony Seabrook. And we also have a panel session including some practicing farmers and others. So I certainly encourage you to get the QR code and look at it. And there's already a number of people have reserved their seats. It will be at the University Club Auditorium, and it will be fully catered and no charges. So please do spread around, particularly students and early career researchers should come. The next one is the Shendon Park Open Day, that is on the 13th of September. And this event is normally done once in 3 years or 2 years. We have the Minister for Agriculture, hopefully will come and launch that. And the things which you can see there, for example, gene-edited wheat breeding. Gene editing is a very hot topic, and that is being grown by the Intergrain, the aquaculture facility, coastal and offshore research lab, canola breeding led by Wallace Cowling and team, the Center for Engineering Innovation, that is agriculture and ecological restoration, then of course the pasture legume breeding program based there. So there's quite a lot to see, and please follow the instructions. So once more, please congratulate the students again for their wonderful presentation. Professor Amanda Davies is already converted. She's going to come and work for us, so she's here again. Thank you, Amanda. I think Abbie has to go, but we should thank Abbie Rogers, Associate Professor Abbie Rogers, then of course Patrick Finnegan, and my team at the Institute. I don't do much work. So Rosanna Cora is not here, still working there. Then I have Diana here, and also my two associate directors, Phil Werko and Wallace, is having a good time in Sydney. So he said, oh, you should change this postgraduate showcase because he wants to be here. I said, we can't change that. So once again, thank you everyone for your patience and asking the questions. There will be more. You can interact with those students. Then we'll have now some drinks and nibbles in the Bayliss foyer. So thank you again. Officially, I close.